Muy buenas tardes. A nombre del Colegio Nacional en general, a nombre en particular del doctor Eusebio Juaristi, del doctor Bolívar y del mío propio, les quisiera dar la bienvenida a las actividades que vamos a tener durante toda esta semana para discutir el mundo del RNA, no solamente como una fase que reconocemos hoy en día como muy antigua en la evolución biológica, sino también como una molécula y una serie de procesos que siguen jugando un papel absolutamente central en la biología contemporánea. Como todos ustedes saben, tenemos un programa que comienza hoy con 2 charlas por día, que culminan el próximo lunes con la presencia de la doctora Ada Jonat del Instituto Weizmann de Israel, premio Nobel en Química en el 2009, y que nos va a hablar sobre los ribosomas, sobre la estructura terciaria de los ribosomas. Yo quisiera decir que, como ya lo han notado algunos colegas del Colegio Nacional, siempre es un placer enorme ver que en las charlas que están relacionadas con ciencia tenemos una enorme cantidad de estudiantes de licenciatura y de posgrado, y algo que nos da especialmente mucho gusto a los doctores Bolívar, Juaristi y a mí mismo es el hecho de que el colegio se está convirtiendo en un punto de reunión para estudiantes que vienen de la UNAM, del Politécnico y de la UAM, como debe de ser. Esta es su casa. Dicho esto, pues vamos a proceder con una primera introducción muy rápida de lo que es el mundo del RNA, y le cedo la palabra a la doctora Negrón.
Bueno, buenas tardes. Para mí es un gran honor iniciar la presentación de las conferencias de esta tarde y que prometen ser muy interesantes, al igual que las que se van a llevar a cabo a lo largo de esta semana. Y por eso quiero agradecer a los organizadores del evento, al doctor Francisco Bolívar Zapata, el doctor Antonio Lascano Araujo y el doctor Eusebio Juaristi, miembros del Colegio Nacional y que es el patrocinador de este evento. Por esta iniciativa tendremos connotados investigadores en el fascinante mundo del RNA, al cual han dedicado mucho de su esfuerzo y tiempo en su investigación. Así, el RNA será nuestra estrella en esta semana, ya que esta molécula se sitúa en un papel central en el origen de la vida, lo que ha originado numerosos estudios de las funciones del RNA que se desconocían. El DNA y las proteínas parecían ser las moléculas dominantes en las células vivas y, por ejemplo, aún en los años 70, 80, en las reuniones, por ejemplo, de ISOL, que es la Sociedad Internacional para el Origen de la Vida, había un enorme debate si primero eran las proteínas o primero era el DNA. Y no se mencionaba mucho al RNA. El mundo del RNA es una solución para esta paradoja. La historia sobre la investigación del RNA, yo lo asocio al cuento de Hans Christian Andersen, que es el patito feo. El patito feo tuvo una evolución de un pato torpe, grande, diferente de sus hermanos y que evolucionó a un cisne muy hermoso. Así, las características fisicoquímicas del RNA permiten proponer a esta molécula como capaz de servir tanto de transmisor, de almacén de información, así como de catalizador, en base de la síntesis de lo que los ribonucleótidos precedía a los desoxirribonucleótidos, la síntesis de proteínas no puede ocurrir sin RNA y muchos cofactores enzimáticos tienen su origen en un ribonucleótido. El descubrimiento de los ribosomas en 1982 proporcionó una base experimental de la capacidad catalítica del RNA, lo que impulsó la universalización de la hipótesis del mundo del RNA como puente entre el mundo prebiótico y el mundo vivo, o sea que era una molécula primordial para la evolución de la vida. Abundan numerosas dificultades para la hipótesis del mundo del RNA, pero sin embargo cada vez se conoce más. Y durante esta semana tendremos la posibilidad de conocer más sobre estos aspectos del fascinante mundo del RNA. Esta tarde tengo el honor de presentar primeramente a Antonio Lascano Araujo, que es miembro del Colegio Nacional y que va a ser el primero de los expositores. Una pequeña referencia de lo que ha hecho Antonio, ya que ha hecho muchas cosas, es que realizó sus estudios de licenciatura y de doctorado en la Facultad de Ciencias, donde actualmente labora fundando el Laboratorio de Origen de la Vida y la asignatura Origen de la Vida, que es una de las matrículas más altas en la facultad. Él se ha dedicado por más de 35 años al estudio del origen y de la evolución temprana de la vida y ha sido profesor visitante en numerosas universidades. Tiene muchas publicaciones en revistas de mucho prestigio, en particular Nature y Science. Es autor de 3 libros en español. Uno de ellos es el bestseller sobre el origen de la vida, con el cual muchos de nuestros jóvenes se han iniciado en este tema. Ha sido galardonado en muchas ocasiones y quiero destacar nada más 3 distinciones recientes, como ser miembro del Colegio Nacional, el Premio Universidad Nacional y más recientemente su nombramiento como profesor emérito de nuestra Casa de Estudios. Forma parte de comités de organizaciones científicas y es el único latinoamericano que ha sido presidente de ISOL, esta asociación a la que me refería anteriormente. En lo personal, algo que me gusta mucho en las conferencias de Antonio es las alusiones que hace a alguna pintura, una foto, una anécdota, y que las asocia de forma muy amena y eficientemente al contexto de que está desarrollando. Y entre otras cosas demuestra pues una gran cultura y también esa característica de ver más allá de lo evidente. Por ello, y por muchas otras cualidades, como ser humano que no son tan conocidas, es para mí un honor contar con su amistad de muchos años, que se inició de una manera muy singular con una visita de au pairing que Antonio organizó en la Facultad de Ciencias. Pues por eso vamos a oír a Antonio en el mundo del RNA, pasado y presente. Te cedo la palabra.
Muchas gracias. Muchas gracias. Tengo que comenzar diciendo que la doctora Alicia Negrón, que es una amiga muy querida, no me dejó ver la presentación antes, porque si no, no le hubiera yo permitido elogios tan desmesurados. Pero muchísimas gracias, Alicia. Bien, lo que yo voy a hacer es darles una introducción muy breve a por qué creemos que el RNA es una molécula tan central en el estudio del origen de la vida y, como se mencionó en un principio, a distintos procesos biológicos contemporáneos. Y lo primero que hay que comenzar diciendo es que, como muy bien decía la doctora Alicia Negrón, el RNA siempre fue el patito feo de la biología molecular. Muchos de los que estamos aquí nos tocaron clases en donde se decía, bueno, el RNA es el mensajero y es el intermediario, y pasemos al siguiente capítulo, que es o la replicación del DNA o la síntesis de proteínas. Y de hecho, lo que hay que decir es que también solemos no festejar el origen de los estudios del RNA, porque no sabemos exactamente en qué momento ubicarlos históricamente. Sabemos muy bien cuándo fue descubierto el DNA, sabemos que hubo gente que con una intuición biológica tremenda a finales del siglo XIX, comienzos del siglo XX, comenzaron a sospechar que el DNA podía ser el material genético celular, y desde luego con el modelo de la doble hélice del DNA de Watson y Crick en 1953, el DNA brinca de inmediato a la palestra de no solo de las ciencias de la vida, no solo de la biología, sino de la ciencia en general, y se convierte en un ícono de lo que puede ser nuestro conocimiento, nuestra descripción secular de los procesos biológicos. El RNA, de hecho, fue visto de una manera siempre secundaria, y los que conozcan a William Shakespeare recordarán que hay una escena muy famosa en donde están Romeo y Julieta en el balcón, y claro, uno es Capuleto, la otra es Montesco, o al revés, siempre me confundo, y de repente ella le dice, ¿por qué tú eres Montesco y yo soy Capuleto? ¿Por qué no nos podemos amar? Pero se los dice con un verso precioso de William Shakespeare, que es What's in a name? ¿Qué es lo que hay en un nombre? Bueno, en el caso del RNA sabemos exactamente lo que hay en el nombre. El RNA, como vamos a ver a lo largo de toda esta semana, y esto es esencial tenerlo presente, es un polianión porque se alternan los grupos fosfato con los azúcares, la ribosa, y los fosfatos tienen desde luego carga negativa, lo cual le permite ser soluble en el agua, y es un ácido nucleico en donde se alternan desde luego la ribosa con el fosfato, ribosa, fosfato, etcétera. Y todos los que están aquí saben muy bien que osa es la terminación que indica que se trata de un azúcar, y RIB son las iniciales del Rockefeller Institute of Biochemistry, que es donde el RNA fue caracterizado por primera vez y fue desdeñado, porque la gente decía, los pobres 3 o 4 investigadores que trabajan en esta molécula no se dan cuenta lo tediosa que es, lo aburrida que es, con solamente 4 bases, y que no parece tener mayor implicación biológica. No es sino hasta los años 50 cuando comienza a desarrollarse ya el estudio molecular de una serie de estructuras como los virus, cuando en un grupo absolutamente excepcional, donde estaba John D. Bernal, que incluía a John D. Bernal, el profesor de Crick, el profesor de Rosalind Franklin, el autor de estos libros un poco stalinistas, pero es un reflejo de la época, de la historia de la ciencia, de la sociología de la ciencia, el amigo de Picasso, etcétera, se dan cuenta que hay virus como el virus del mosaico del tabaco, que tiene RNA como material genético. Y como en esa época se creía erróneamente a que los virus, por ser muy sencillos y por poderse cristalizar, eran primitivos, se llegó a una conclusión completamente correcta a partir de premisas un poco cuestionables. Como algunos virus, como el del mosaico del tabaco, poseen genomas de RNA, y como se pueden cristalizar, lo que algunos creían que indicaba que estaban presentes en la transición de lo vivo y lo inerte, entonces, ¿cuál fue la conclusión? Que los genomas de RNA deben ser primitivos. Conclusión correcta, o como dirían los ingleses, un reloj descompuesto a veces indica la hora correcta, cuando menos una vez a lo largo del día. Bueno, esto fue fascinante, pero cuando uno revisa la historia de los años 50, de los años 60, es absolutamente deslumbrante en términos de cómo fuimos avanzando en términos del conocimiento de los procesos básicos de la célula. Y no se olviden ustedes que una vez propuesto el modelo de la doble hélice del DNA, la pregunta es, ¿y cómo es que están codificados en el DNA las secuencias de los aminoácidos de las proteínas? Y en esta diapositiva de un libro espléndido que me acaba de regalar el día de ayer mi amiga María Cristín Moguel, pueden ver ustedes esta especie especie de caja negra, en donde la gente sospechaba debería estar algún tipo de molécula que fungiera como intermediario entre el DNA y las proteínas, pero realmente no había ninguna idea de qué era lo que estaba ocurriendo. Pero haciendo gala de esa intuición biológica deslumbrante, absolutamente extraordinaria, Francis Crick empezó a sospechar que los datos disponibles indicaban que el intermediario tendría que ser precisamente el RNA. Esto va a coincidir muy rápidamente con una revisión crítica que va a hacer Crick de cuál es el proceso de transmisión de la información genética y de la replicación de la información biológica, y esta página que está tomada de la red les muestra en los apuntes originales del artículo que escribe Crick en 1958, en donde no solamente va a decir que las proteínas son depositarias de la historia de los seres vivos, sino también va a decir que el RNA claramente es un intermediario en la transición de la información genética del DNA hacia las proteínas, y esto es lo que él va a llamar el dogma central. Las flechitas las dibujó Crick, y algunos de los que estamos aquí conocimos a la secretaria de Crick que todavía hizo a máquina esta página. Aquí hay que enfatizar, sobre todo para aquellos que tienen una interpretación a veces un poco peculiar de la epigenética, que lo que Crick está diciendo es que la información nunca fluye de las proteínas hacia el RNA o hacia el DNA, sino que siempre la información tiene una cierta dirección. Pero aquí queda ya perfectamente establecido el papel del RNA como un intermediario en los procesos genéticos, en los procesos de expresión de los genes. Al mismo tiempo, se estaba llegando al momento culminante de su desarrollo una disciplina que desafortunadamente hemos olvidado, que era la bioquímica evolutiva. Durante los años 30, 40, 50, hasta finales de los años 60, hubo un grupo deslumbrante de científicos a ambos lados del Atlántico que se preocuparon por interpretar los fenómenos bioquímicos desde una perspectiva evolutiva. Y la idea que llevó a muchos a fijarse en el RNA era el hecho de que si nos fijamos en este diagrama de Camembert, que no importa que sea moderno, porque eso en realidad se conocía desde los años 50, la mayor parte del peso seco de una célula está constituida por proteínas. Tenemos más o menos una cuarta parte del material que está formado por fosfolípidos, coenzimas, poliaminas, etcétera, pero una buena rebanada de este queso Camembert, como se dan ustedes cuenta, está formada por RNA. Y otra vez, haciendo gala de una intuición maravillosa, algunos personajes como Brachet en Bélgica, como Belozersky en la Unión Soviética, se dan cuenta que hay tanto RNA, tan poco DNA, pero tanto RNA, que llegan rápidamente a una conclusión, puesto que el RNA es el segundo componente más abundante en las células, seguro tiene que ser importante. Y empiezan a percatarse por primera vez de que efectivamente algunas coenzimas, sin las cuales no podemos tener actividad enzimática, sin las cuales las proteínas que son enzimas no pueden funcionar, algunas coenzimas son derivados de ribonucleótidos, y se dan cuenta de que paralelamente al proceso de división de una célula tenemos la replicación del DNA, pero si no está presente el DNA, el RNA, eso no puede darse. Y entonces, en un acto absolutamente de audacia intelectual, posible porque para entonces Stalin ya había muerto, se había muerto en 53, y ya la biología molecular se empieza a desarrollar en la Unión Soviética de una forma espléndida. Belozersky, que era un estudiante de Oparin y que por lo tanto había crecido desarrollando, viendo la idea de que los procesos bioquímicos eran susceptibles de ser interpretados evolutivamente, Belozersky, en un congreso que hay en Moscú en 57 y que se van a publicar las memorias en 59, escribe diciendo: no hay ninguna duda de que los ácidos nucleicos jugaron un papel importante en la evolución del mundo orgánico y en las reacciones metabólicas. Sin embargo, es difícil pensar que el RNA y el DNA hubieran podido surgir simultáneamente en la evolución temporal. temprana de la vida. Más bien parece que los ribonucleótidos y luego el RNA se originaron primero. El DNA apareció mucho más recientemente a medida que el protoplasma se hizo más diferenciado y sus funciones se desarrollaron, aumentaron en complejidad. Pero vean aquí todavía la permanencia del término protoplasma, que produce sentimientos de añoranza por lo inexacto de lo que del término, pero la maravilla que refleja este proceso precisamente de incremento de nuestro conocimiento de los fenómenos celulares. Parece que el RNA, al estar asociado con los procesos más generales de la vida, surgió en una etapa evolutiva más temprana, mientras que el origen del DNA está asociado con el desarrollo de rasgos más especializados y filogenéticamente tardíos de las células. Aquí lo que estamos viendo eran científicos que eran formados en química, en biología celular, en la naciente biología molecular y desde luego en una perspectiva evolutiva, y tenían una visión que debe ser un aprendizaje de cómo debemos enseñar las ciencias de la vida en nuestros días. Paralelamente a estos descubrimientos y de manera totalmente independientes, Philip Handler, que fue un bioquímico absolutamente extraordinario estadounidense, fue presidente de la National Academy of Sciences, un extraordinario promotor de los intercambios académicos entre los distintos países en plena época de la Guerra Fría, Akin, que trabajaba en la Universidad de Austin, Leslie Orgel en 68 primero y luego en 71, y Harold White en 1976, se ponen a ver la actividad catalítica y se dan cuenta que las enzimas solo pueden funcionar si tienen asociadas coenzimas y las vitaminas, como todos sabemos, ¿no? Las vitaminas, pero que además buena parte de estas coenzimas son precisamente derivados de ribonucleótidos y en particular derivados de la adenosina. Aquí pueden ver la adenina, ribosa, fosfato, adenina, ribosa, fosfatos, adenina, ribosa, fosfatos, etcétera. Y esto, con una audacia intelectual verdaderamente extraordinaria, los lleva a pensar que tal vez en las coenzimas estamos viendo los vestigios de una época primordial en que la catálisis biológica, las rutas metabólicas, no dependían de proteínas, sino dependían de ribonucleótidos o de moléculas un poquito más complicadas, pero que tuvieran coenzimas asociadas, y a partir de allí se desarrollan procesos que luego llevarán a la aparición de las enzimas. De todos estos autores, probablemente el más refinado, el más sofisticado, sea el artículo de Leslie Orgel de 1971 y el de Harold White. Pero aquí permítanme enfatizar, sobre todo en beneficio de los jóvenes que están aquí, esta capacidad para percibir fenómenos a gran escala y ver algo, proponerlo con mucha audacia. Aunque también hay que decir que cuando Handler escribió su artículo, él dijo, pues yo no sé nada de coenzimas, no sé nada de la evolución de coenzimas, pero me puse a pensar al ver las reacciones bioquímicas y quiero proponer esta idea, lo cual es de una espontaneidad absolutamente maravillosa. Pero entonces se dan ustedes cuenta cómo había ya estos indicadores sobre la importancia del RNA y los ribonucleótidos. El doctor Michael Jarvis va a abundar precisamente en su charla sobre este tipo de procesos. Para los años 60 ya era perfectamente claro que uno puede tener síntesis de proteínas sin necesidad de DNA. Necesito el RNA mensajero, necesito el ribosoma, que es una estructura básicamente compuesta por RNA y proteínas asociadas, y aquí vamos a tener algunos de los grandes especialistas en el estudio de los ribosomas, pero además la gente se empezó a fijar en la estructura del RNA de transferencia. Todos ustedes conocen el proceso de síntesis de proteínas, el mensajero se asocia al ribosoma, llegan los RNAs de transferencia cargados, se forma el enlace peptídico en el sitio de la peptidiltransferasa en el interior de la ribosoma, pero algunos, otra vez, dotados de esta intuición, de esta visión tremenda, se dan cuenta que la estructura terciaria del RNA de transferencia es prácticamente tan compleja como la de una enzima. Y aquí, de nueva cuenta, se queda uno asombrado de la intuición de Francis Crick, que con mucha naturalidad, en 1966, escribe, como se ha señalado en otros lados, el RNA de transferencia parece como un intento de la naturaleza para hacer que un RNA haga el papel de una proteína. proteína. Y eso estaba basado en la estructura, estaba basado en el reconocimiento de la complejidad estructural del RNA de transferencia. Y luego, en 1968, en un artículo que Crick escribe al lado y publica al lado de un artículo de Leslie Orgel en el Journal of Molecular Biology, sin que ambos hubieran comparado sus ideas. Eran amigos de mucho tiempo, pero no habían comparado las ideas que estaban expresando finalmente en ese artículo. Lo que va a escribir Crick dice, una idea muy atractiva que nos ha sugerido el doctor Oliver Smithis es que el RNA de transferencia primitivo era su propia enzima activadora, es decir, que la estructura tenía una cavidad en la cual de una manera muy específica podía colocarse la cadena lateral del aminoácido correspondiente en una posición tal que el grupo carboxilo del aminoácido pudiera fácilmente unirse al azúcar terminal del RNA de transferencia. Es decir, lo que están ustedes viendo aquí es muy claramente la capacidad de Crick para decir si la estructura terciaria de la molécula es tan compleja, eso me permite predecir que son moléculas con actividad catalítica, porque la actividad catalítica de las enzimas ciertamente depende de su estructura terciaria. Bien, esto es lo que va a llevar a que otra vez de manera independiente Alex Rich en 62, Woese en 1967, Crick en 68 y Leslie Orgel en 68 propongan lo que ahora conocemos como la hipótesis del mundo del RNA, con la idea de que en la Tierra primitiva había RNAs que se podían replicar, que estaban codificando para RNAs catalíticos que estaban a su vez permitiendo que se replicara más RNA. Aquí permítanme subrayar un punto que es central en la biología. Para los físicos newtonianos es muy fácil hacer predicciones. Para los químicos es fácil hacer predicciones de cómo puede ir una reacción, más o menos, a veces falla. Pero en biología es muy difícil hacer predicciones con esta precisión. Lo que Crick, Orgel, Woese y Rich estaban prediciendo en realidad, estaban haciendo la predicción de que el RNA podía ser catalítico. ¿Cuál es el origen de este trabajo tan excepcional, de esta visión tan excepcional? En primer lugar, la intuición biológica. Conocían perfectamente los procesos que estaban estudiando, los procesos que estaban describiendo, conocían muy bien las estructuras de las moléculas, había un marco de referencia evolutivo, cosa que después muchos biólogos moleculares abandonaron, aunque venturosamente eso se ha recuperado, y finalmente conocían perfectamente bien la biología molecular porque eran los fundadores. Había un elemento adicional que a mí me gusta mucho enfatizar y era el intercambio extraordinario de ideas que había entre ellos. Leslie Orgel y Francis Crick siempre fueron muy amigos, pero si ustedes se meten a los archivos que seguramente deben estar en la red de la correspondencia de Orgel, de Crick, van a ver la libertad con la que cambiaban las ideas con Woese, con Alex Rich, y en realidad en esa época la comunidad era pequeña y se veían no como competidores, no estaban compitiendo por recursos, por espacio, por estudiantes, sino que estaban conscientes que estaban haciendo nacer una nueva forma de ver la biología. Desafortunadamente, la situación ha cambiado y muchos de los que ahora estamos en ciencia ya no tenemos colegas, sino competidores. Pero en este momento había objetivos intelectuales muy claros y había una visión de grupo que, de nueva cuenta, creo que es una lección que deberíamos tener en mente. Bueno, lo que realmente estaban diciendo Crick y Orgel, Woese y Rich, pero sobre todo Orgel, creo que es el artículo más refinado, es que ciertamente el DNA se puede replicar con el concurso de las proteínas tienen propiedades estructurales, catalíticas, gracias a la estructura terciaria que tienen y están codificadas por el DNA, pero lo que es la idea básica del mundo del RNA es que antes que viniera el DNA y antes que vinieran las proteínas, vino en realidad el RNA. Esta es la idea básica de la hipótesis que están sosteniendo de manera independiente y de nueva cuenta yo creo que la propuesta más refinada es precisamente la de Leslie Orgel. Bueno, ¿qué fue lo que pasó en los 20 años que siguieron a la propuesta del RNA como la molécula primordial? Hasta sus mismos autores la hicieron a un lado. hasta sus mismos autores, como vieron que muy poca gente les hacía caso, dejaron de defender en público al menos la idea de que el RNA había precedido al DNA y a las proteínas, y en los 20 años que siguieron a las publicaciones de Crick y Orgel, por ejemplo, casi todo el mundo aceptaba la hipótesis de que los genomas de RNA vinieron antes que los genomas celulares de DNA. Esto lo apoyaban Liebman, Haldane, Oparin, Belozersky, muchos autores sin ningún problema. Muchos aceptaban la posibilidad de que las coenzimas eran vestigios de un mundo de RNA, pero pocos estaban trabajando en esta idea. Algunos aceptaban la idea de que la histidina era un vestigio molecular de una etapa del mundo del RNA, pero la primera que recupera esta idea es precisamente Marie-Christine Morel, que está con nosotros, que va a dar una charla el día de mañana, cuando hizo su tesis y convenció a Jacques Nignot, que no era una proeza fácil de trabajar en este punto. Se desarrollan los métodos a partir del extraordinario trabajo de Sol Spiegelman para lograr in vitro la evolución de sistemas de RNA, trabajando con el virus de la el virus cubeta y la cubeta replicasa, pero la conclusión es que nadie estaba realmente trabajando en las propiedades catalíticas del RNA, hasta que de repente, auténticamente, por casualidad, se descubre en dos laboratorios, en el laboratorio de Tom Sheck y en el laboratorio de Sidney Altman, que el RNA es catalítico. Y para que vean ustedes hasta qué punto la gente se había olvidado de esta posibilidad, déjenme decirles que en esos momentos estaba yo haciendo unos experimentos en el laboratorio de Joan Oro en Houston. Apareció el artículo publicado en Cell donde reportaban la actividad catalítica del RNA. Fui a ver a Joan Oro, que era uno de los grandes investigadores en origen de la vida, en química prebiótica, le enseño el artículo, y Joan, que llevaba toda su vida trabajando en RNA, ve el artículo y me dice, bueno, pero aquí lo único que tenemos es actividad hidrolítica, cualquier molécula puede tener actividad hidrolítica, y la desdeñó. Con otros no ocurrió lo mismo, en particular con el grupo de Sidney Altman. En 1983 publican un artículo en donde dicen, si las proteínas fueron entidades que arribaron relativamente tarde en la evolución de las macromoléculas, entonces las primeras manipulaciones de los ácidos nucleicos deben haber sido llevadas a cabo enteramente o predominantemente por ácidos nucleicos catalíticos. Los vestigios de estos eventos biológicos importantes tempranos pueden ser aparentes en la reacción de la RNA-SAP, que era la molécula en la cual siempre trabajó Sidney Altman, en el procesamiento de algunos RNAs y en los ribosomas, y sin lugar a dudas en otras reacciones catalizadas por RNA que aún permanecen por ser identificadas. Bueno, resulta que en estas épocas alguna vez se me ocurrió preguntarle tanto a Leslie Orgel como a Francis Crick en una de esas comidas memorables en las que uno tenía la posibilidad de ser invitado en Piatis, un restaurante en La Joya que debería ser declarado monumento de la biología molecular en los Estados Unidos, y les pregunté, ¿y cómo ven el descubrimiento, la corroboración de que efectivamente el RNA es catalítico? Las respuestas son fascinantes, porque Leslie Orgel me dijo, si yo hubiera sabido que aún había actividades catalíticas del RNA, hubiera modificado toda mi carrera científica y la hubiera dedicado al estudio de las moléculas de RNA. Y Francis Crick, con un sentido de humor muy inglés, me dijo, ya se me había olvidado que había propuesto esa idea. Por supuesto que no se le había olvidado, pero la gente no creía en eso porque todos habíamos sido educados en el dogma de que en la biología la catálisis depende del RNA, de las proteínas, y los ácidos nucleicos almacenan información genética. Se dan ustedes cuenta la restricción tan brutal, el freno intelectual tan brutal al que estábamos sujetando a la biología. E incidentalmente, déjenme decirles que una de las motivaciones para hacer toda esta semana es el hecho de que en México, lamentablemente, en nuestros cursos de biología celular y molecular, seguimos sin darle la atención debida a la biología molecular del RNA y a las moléculas y a su actividad catalítica como debiéramos. Pero déjenme ponerles un ejemplo que demuestra cómo esta visión puede ser muy parcial. Miren, aquí esta tabla la preparó Ricardo Hernández, que está sentado aquí, es un colega, un estudiante de laboratorio, y ven ustedes la catálisis biológica, cómo puede ser llevada a cabo por enzimas o por ribosomas. ribosimas, por RNA catalíticos. Y aquí ven en esta primer columna la clasificación de las distintas enzimas según la Comisión Internacional, la Unión Internacional de Bioquímica. Tenemos la Enzyme Commission Number 1, Enzyme Commission Number 2, Enzyme Commission Number 3, y ven oxioreductasas, transferasas, hidrolasas, liasas, isomerasas y ligasas. Y en la segunda columna ven ejemplos que muchos de los que están aquí conocen de las actividades que estas distintas enzimas catalizan, las actividades bioquímicas, dependiendo del grupo al que pertenezcan. Pero ven en la última columna cómo hay ribosimas, RNAs catalíticos, que llevan a cabo exactamente el mismo tipo de reacciones. Es decir, si queremos entender la catálisis biológica, en modo alguno podemos darnos el lujo de pretender las ribosimas, y es absurdo que no hayamos logrado promover esta visión en la enseñanza de la biología entre los jóvenes que están estudiando ciencias de la vida. Bueno, desde luego que el ejemplo más notable de catálisis biológica, y de esto va a hablar primero Lauren Williams, después va a hablar Ada Jonath, es el ribosoma. Si ustedes se fijan en esta imagen que viene precisamente del laboratorio del doctor Lauren Williams, pueden ver ustedes cómo las proteínas ribosomales están pegadas afuera de las subunidades grandes y pequeñas del ribosoma, y pueden ver cómo en realidad el sancto sanctorum, el sitio donde se lleva a cabo la formación del enlace peptídico, es precisamente un sitio donde no tenemos proteínas, solamente tenemos RNA, que forma estas extraordinarias estructuras de una complejidad espacial enorme. Hasta aquí uno podría decir, bueno, esto valida desde el punto de vista del pasado las actividades del RNA y los esfuerzos que podamos hacer para tratar de estudiarlos. Pero déjenme tratar de mostrarles hasta qué punto de veras es importante el RNA. Miren, si yo quiero tener RNA, lo que necesito es juntar ribonucleótidos con una enzima que se llama RNA polimerasa. Y si yo me fijo en los ribonucleótidos, todos son sintetizados en una serie de pasos enzimáticos comunes a los seres vivos, lo cual quiere decir que aparecieron seguramente muy tempranamente en la evolución, que son de origen monofilético, a partir de una serie de precursores, algunos de los cuales sabemos cómo pudieron haber estado en la Tierra primitiva, pero otros realmente, como las ribosas fosfatadas, no tenemos una buena explicación. Bueno, una vez que tengo ribonucleótidos y tengo el RNA puede estar dividido en dos grandes grupos: el RNA codificante, como por ejemplo el que vemos en el virus del Zika, el que vemos en el RNA mensajero, el que vemos en los viroides, sin ningún problema. Y como todos ustedes saben, el RNA tiene una propiedad que no vemos en el DNA, que son los riboswitches. El RNA logra al mismo tiempo controlar la expresión del mensaje genético que llevan. Luego podemos tener en los RNAs no codificantes, podemos tener los pequeños RNAs no codificantes, como todos los sistemas de regulación, que prácticamente cada mes estamos descubriendo nuevas formas de regulación en donde el RNA juega un papel central. Lo mismo en el caso de la diferenciación celular, que en la expresión de proteínas en las plantas, que por ejemplo en la diferenciación tisular del cerebro de los cefalópodos, de manera que nos damos cuenta del extraordinario papel que el RNA también juega como regulador. Podemos tener moléculas grandes no codificantes de RNA, como el RNA ribosomal, el de transferencia o los primers. Y luego, en lo que es realmente una demostración de la validez de las premisas de la bioquímica evolutiva, es si yo quiero tener DNA, necesito desoxirribonucleótidos, y en absolutamente todas las células, para tener desoxirribonucleótidos, necesito una reacción universal, que es la reducción de los ribonucleótidos. Eso es lo que me está diciendo, en una interpretación muy obvia, muy clara, muy directa de un proceso bioquímico, es que los ribonucleótidos antecedieron a los desoxirribonucleótidos, la idea de Belozersky de que el RNA vino antes que el DNA. Y luego finalmente, y esto es algo de lo que nos ocupamos mucho en el laboratorio, muchos ribonucleótidos pueden ser coenzimas alarmonas o histidinas, algo que hemos estado estudiando en el laboratorio. Ese es el trabajo de tesis de Alberto Vázquez, de Betunes Betabeles, junto con Arturo Becerra yo mismo, con algunos miembros del laboratorio. La histidina, en efecto, es una molécula que se forma usando como precursor ATP. No existe ningún otro aminoácido que tenga esta propiedad. Es el aminoácido más abundante en los sitios catalíticos de las enzimas, de manera que lo que la biología me está diciendo es: ¿quieres entender el origen de la catálisis biológica? Explica el origen de la histidina. Y peculiarmente, singularmente, es el único aminoácido que comienza su síntesis a partir de ATP. Tenemos desde luego las alarmonas, que son toda esta gama de mensajeros secundarios que hemos estado estudiando en el laboratorio como parte del trabajo de Ricardo Hernández, que está por aquí, junto con Arturo Becerra, y aquí puse solo una de ellas, pero tenemos una gama de alarmonas y todas son ribonucleótidos o ribonucleótidos modificados. Y finalmente, y de esto va a hablar el doctor Yarrus, tenemos las coenzimas que, como ya vimos, son absolutamente críticos en la actividad catalítica de las enzimas. De manera que uno puede decir que el RNA es fascinante, pero los ribonucleótidos son monómeros de RNA, son moléculas asociadas con los procesos energéticos, son precursores de los desoxirribonucleótidos, son coenzimas o precursores de coenzimas, son alarmonas o precursoras de alarmonas y son precursoras de la histidina, que es el aminoácido más crítico en la catálisis biológica en términos generales. Con todo eso, ¿cómo podemos definir el mundo del RNA? Bueno, yo creo que existen muchas definiciones del mundo del RNA, incluyendo varias que son contradictorias, pero yo estaría perfectamente satisfecho con decir que las propiedades catalíticas, reguladoras y estructurales de las moléculas de RNA y los ribonucleótidos, combinados con su ubicuidad en los procesos celulares, sugieren que es una etapa temprana, tal vez primordial, durante las cuales las moléculas de RNA jugaron un papel mucho más conspicuo, mucho más visible en la herencia y en el metabolismo. Hay quienes difieren de esta opinión. En toda justicia debo decir que hay quienes creen que un mundo de RNA nunca existió. De acuerdo, dejemos lo que se estén cocinando en su herejía. Pero si eso es el caso, lo que tienen que explicarnos entonces es por qué cuál es el origen y los múltiples papeles del RNA, porque es una molécula tan ubicua en los procesos biológicos. Y la interpretación no solo más parsimoniosa, sino más lógica es reconocer que estamos viendo las derivaciones de un proceso muy antiguo. Bueno, para terminar, quisiera decir entonces que una hipótesis de trabajo que es extraordinariamente fácil de aceptar es que hubo de alguna manera una sopa primitiva. No sabemos bien a bien cómo ir de esta sopa primitiva a un mundo del RNA. Aquí tenemos a uno de los grandes expertos en esta transición o en los problemas que están presentes, como es el doctor Ram Krishnamurti, y todo indicaría que la síntesis de proteínas surgió en un mundo de RNA y que el DNA apareció tardíamente en la evolución. Y déjenme poner un ejemplo extraordinariamente cercano al Colegio Nacional, que es el Templo Mayor. Si ustedes van al Templo Mayor, ustedes saben que lo que estamos viendo son los vestigios de una enorme pirámide, que era una estructura muy bella, muy compleja, extraordinariamente refinada desde el punto de vista arquitectónico, estético, funcional. Pero la pirámide que alguna vez adornó el Zócalo, el Templo Mayor, en realidad fue construida por etapas. Y lo que hacen nuestros amigos arqueólogos, lo que pueden ver ustedes, es cómo hay las demostraciones estructurales de que hubo pirámides más pequeñas, más pequeñas, más pequeñas, hasta una pequeña pirámide original. La idea central del mundo del RNA es que esta pirámide original pudo haber sido la molécula del RNA. Pero como todos ustedes saben, para sostener las piedras necesitamos estuco, necesitamos baba de nopal, necesitamos distintos tipos de cemento que las culturas mesoamericanas desarrollaron, de manera que en lo que no podemos pensar es en un mundo donde el RNA estuviera puro sin el concurso de muchas otras moléculas. Esto complica nuestra descripción de lo que ocurrió en la Tierra primitiva, ciertamente, pero al mismo tiempo lo vuelve un reto intelectual absolutamente fascinante, un problema abierto en el que todos, sabiendo la biología molecular, conociendo la estructura de las moléculas, podemos asomarnos y hacer una serie de predicciones o llegar a una serie de conclusiones. Muchas gracias.
doctor Lascano, y ven como si tenía razón, ven más allá de lo evidente, con la pirámide, ahí me lo demostraste. Tenemos tiempo para preguntas, por favor, si alzan la mano. Yo no veo. A ver, ¿por allá? Sí.
Sí, para mí el tema es muy complejo. Yo no estoy metido en la biología, pero sí estoy metido en la parte sociológica. A mí me surgen muchas dudas, por ejemplo, hablando del Templo Mayor. En el Templo Mayor se ha encontrado cráneos, esqueletos, bla, bla, bla. Hay una infinidad de hallazgos muy importantes, pero simple y sencillamente nos quedamos ahí. O sea, no tenemos más investigación de esos grupos originarios de México. Pudieron tener contacto con los africanos, es decir, en DNA, o pudieron esto, lo otro. O sea, estamos todavía como muy abajo en las teorías. Aparecieron los aztecas y ahí nos quedamos, pero no sabemos todavía cuál es el origen de estas razas, de nuestras razas de México. Y creo que también hay una beta muy grande cuando esto lo clavamos a la parte arqueológica. Y creo que la duda no es en México, es prácticamente mundial.
Bueno, la pregunta que usted hace no tiene nada que ver con lo que yo dije, porque nada más me estaba refiriendo a la pirámide como una metáfora de cómo se construyen los procesos biológicos. No quise usar otra metáfora que nos gusta mucho a los biólogos, que es el coxis. El coxis, que en realidad es el vestigio de una cola que alguna vez tuvimos todos los primates y que en el caso de nuestra línea se perdió. No sé qué preguntas si hubiera hecho usted o si hubieran hecho otros, si me hubiera yo referido al coxis en lugar de a la pirámide, pero el punto básico es que así como en la historia siempre tenemos una serie de vestigios que podemos integrar en un gran esquema, en el caso de la biología lo que estamos haciendo es aplicando los principios de Darwin precisamente a nivel celular y subcelular, es que puede uno entender, igual que Darwin entendió que había unas víboras sincrótalo ancestrales a las víboras de cascabel, de igual manera puedo ver en las propiedades del ribosoma lo que alguna vez fue una síntesis de proteínas sin el concurso de enzimas.
¿Otra pregunta?
Hola profesores, buenas tardes. Como siempre un gusto escucharlos. Doctor Lascano, con respecto a la parte como tal del mundo del RNA, ¿qué piensa usted o qué me puede decir con respecto a las teorías de que dicen que es una molécula que no puede soportar tantos cambios en sus medios, ya sean pHs, temperaturas? Muchas personas como que le tiran a la idea de que el RNA no puede ser también una molécula primordial, por así decirlo, antes que el DNA, por cuestiones precisamente de su plasticidad y ese tipo de cosas que de repente meten ruido en ese tipo de teorías.
La plasticidad sería precisamente un argumento a favor de los muchos papeles que la molécula de RNA pudo jugar en la evolución temprana. Respecto a la estabilidad, eso prefiero no tocarlo porque la doctora Morel va a hablar precisamente de cómo se puede jugar con moléculas de RNA que resistan enormes presiones, cómo podemos incrementar la termoestabilidad y respecto a los precursores más estables o menos estables, pero que al mismo tiempo tengan un cierto nivel de reactividad. Esa es la charla que sigue del doctor Ram Krishnamurti.
¿Otra pregunta? Anímense, no tienen siempre tanta oportunidad de tenerlos. Ahí hay una.
Bueno, buenas tardes. Yo recuerdo en mis clases de biomoléculas que mi profesor mencionó sobre los microRNAs en los caldos primitivos. A mí no me quedó muy claro porque no lo enfatizó tan bien, como usted mencionaba que no tienen como ese énfasis en el RNA. pero no me quedó claro en la regulación genética. ¿Cómo actúan los microRNAs en estos caldos primitivos?
Hay una microRNAs en los caldos primitivos, no. Uno puede intentar formar oligómeros de ribonucleótidos en soluciones acuosas, tratando de ver cómo puede crecer la molécula, pero esa es la charla del doctor Uli Müller. Precisamente por eso los trajimos, para que todas estas dudas queden allí.
¿Otra pregunta?
I'm right. Hopefully.
¿Otra pregunta? Allá.
Simplemente quería preguntar, ¿es ya un consenso el mundo del RNA o todavía hay alguna otra visión alternativa reconocida, digamos, en la comunidad de origen de la vida?
Yo creo que si uno examina la ubicuidad y diversidad de los procesos del RNA, la única explicación de los biólogos es que estamos viendo un proceso, las evidencias de una molécula muy antigua que jugó en el pasado un papel muy crítico, pero eso no quiere decir que todos los procesos sean igualmente antiguos. Por ejemplo, los RNAs juegan un papel central en la regulación de muchos procesos en las células vegetales, que ciertamente son uno de los grupos biológicos más tardíos. Entonces, allí hay una plasticidad de la molécula que los seres vivos, con ese oportunismo característico que tenemos, vamos a estar usando en distintas condiciones. Pero yo creo que desde la óptica de los biólogos, yendo del presente hacia atrás, la única posibilidad es reconocer que el RNA en el pasado tuvo un papel mucho más central en herencia y en metabolismo que lo que vemos hoy en día. Lo cual no quiere decir que haya sido la única molécula. Hay que recordar que por muy reduccionistas que sean nuestras descripciones del pasado biológico, siempre tenemos que tomar en cuenta que si había RNA, necesariamente tiene que haber habido aminoácidos, que es muy fácil sintetizar, que si había RNA, seguramente había lípidos, porque sabemos por los experimentos de Dave Dimmer que uno puede extraer moléculas lipídicas de meteoritos y uno puede intentar sintetizarlas en condiciones de tierra primitiva. Entonces hay una gama de moléculas, hay una gama enorme, un espectro grande de especies moleculares, y enfatizamos el RNA porque tiene esta dualidad como material genético y como catalizador, pero en modo alguno quiere decir que reduzcamos los procesos biológicos ancestrales a este tipo de molécula, ¿no? Ahora, hay voces disidentes, pero siempre hay gente que tiene derecho a equivocarse.
Ahí hay un...
Antonio, enhorabuena por tu charla, magnífica como siempre. Respecto a los disidentes que quedan del mundo RNA, como bien decías, quizá uno de las críticas que se solían hacer al modelo de mundo RNA era el propio origen de los ribonucleótidos, porque como es sabido es muy difícil sintetizarlo a partir de los tres componentes por separado. Pero en ese sentido los trabajos del grupo de John Sutherland, su publicación de 2009, nos ha demostrado que se puede formar un ribonucleótido a partir de moléculas muy sencillas y de una forma mucho más eficiente que lo previsto. ¿Qué relevancia le das a este tipo de experimentos, digamos, para fortalecer esta idea del mundo RNA porque nos permiten originar la molécula de una forma mucho más sencilla?
A ver, yo creo que aquí hay que hacer una distinción. No todos los procesos abióticos son procesos prebióticos. Por ejemplo, yo puedo sintetizar en el laboratorio sin ninguna dificultad colesterol, sin problema. Pero el colesterol es una molécula que claramente no estaba presente en la tierra primitiva. Mientras que si hago un experimento como el de Miller, por ejemplo, con todas las desventajas que pueda tener por haber sido diseñado hace 50 años, con modelos más incompletos que los que tenemos ahora del medio ambiente primitivo, y comparo esos compuestos con lo que veo en un meteorito, veo una correspondencia bastante buena en muchos casos. Entonces yo creo que ese es un buen modelo que puede ser refinado de la química de la tierra primitiva. Mi síntesis del colesterol no. Las síntesis que han estado reportando John Sutherland y Michael Powner son absolutamente maravillosas y yo creo que son demostración de un principio. Pero entonces aquí hay un problema que yo, al que no le veo solución. Es Si uno se fija en esas síntesis tan dirigidas del grupo de Sutherland, que son de veras demuestran una habilidad conceptual, técnica extraordinaria, lo que uno ve es una gama muy reducida de compuestos. Ahí está la evidencia empírica. Pero si me voy a los meteoritos, que es la única muestra que yo tengo de la química del sistema solar primitivo, y analizo los compuestos, veo que la síntesis en el sistema solar primitivo eran menos determinísticas. Entonces, lo que John Sutherland nos tiene que explicar, o está proponiendo dos tipos de química distinta, una como la que él está simulando en el laboratorio para sintetizar los compuestos y otra distinta para lo que se da origen a los meteoritos, o la solución debe ser intermedia. Y yo me inclino a pensar en esta segunda opción. Pero como demostración de principios me parece maravillosa.
Sí, doctor, muy buena plática como siempre.
Gracias.
Ahorita siempre ha dado usted evidencias de cómo se sustenta bastante bien el RNA como antecesor del DNA, luego aparecieron las proteínas. No sé si han teorizado cuáles son avances que se tienen, hacia dónde va la evolución molecular. Ya tenemos RNA, ya tenemos DNA, tenemos proteínas. ¿Qué sigue? ¿Cuál sería el próximo salto que habría en este tipo de evolución?
Mire, en evolución uno puede hacer predicciones, explicaciones al pasado, no predicciones al futuro. Decía Jorge Luis Borges en el Manual de la Zoología Fantástica que hay un pájaro gofus que vuela mirando hacia atrás porque no le interesa saber hacia dónde va, sino de dónde vino. Y todos los que hacemos biología evolutiva estamos en esas condiciones. En realidad lo que nosotros podemos hacer, dado el carácter contingente de la evolución biológica, es explicar el pasado, pero no predecir el futuro. Yo creo que ningún químico prebiótico, en el sentido estricto del término, si hubiera estado allí, se hubiera atrevido a pensar que iba a aparecer una ruta tan compleja como la síntesis del triptofano, por ejemplo. Alguna vez Stanley Miller y yo nos pusimos a ver la síntesis del triptofano y dijimos, aquí Dios metió la pata, porque esto iba derechito hacia la formación de un nucleótido, de inosina, pero no fue así. Entonces yo creo que ahí el papel de la contingencia siempre es muy importante y lo vemos en rutas metabólicas, por ejemplo. Hay varias maneras de sintetizar lisina. Hay varias maneras distintas en distintos grupos biológicos. ¿Para qué si ya existe toda una forma de sintetizarla que parecería ser muy eficiente, muy económica? Entonces yo creo que ahí estamos en las mismas condiciones que los historiadores. Explicamos el pasado, pero no predecimos el futuro. Si esto les parece un problema brutal, piensen en que nadie le hace la misma reclamación, por ejemplo, a los economistas, que siempre nos están vendiendo futuros muy dorados y todos sabemos las circunstancias en las que nos encontramos, ¿no?
Acá hay otra.
Muy buenas tardes, doctor Lascano. Primero felicitarle, una ponencia excelente. Me llamo Isidro Ramírez. Su plática me llevó inmediatamente a recordar hace ya unos años un libro que leí que se llama El azar y la necesidad. En ese libro y en aquellas épocas leí la palabra teleonomía, que hablaba pues como de la predestinación de las moléculas y mencionaban escuetamente el RNA. Quizá en ese momento y en esa época no se tenía toda la cantidad de conceptos y de investigaciones y de resultados para entender tanto pues ese concepto de teleonomía y a la molécula de RNA como una molécula teleonómica. Yo quisiera pedirle que quizá me amplíe más ese concepto porque nunca me quedó muy claro qué es la teleonomía y si realmente podemos considerar al RNA una molécula de ese tipo.
No, lo que ahí hay que entender es, el libro de Jacques Monod es una reflexión fascinante sobre un problema que es tan viejo como la filosofía, que son los rangos de libertad que puede tener la naturaleza o que puede tener la mente humana o las sociedades. Y Monod, que tenía esta capacidad extraordinaria para, con una elegancia típicamente francesa, poner los debates filosóficos en un contexto biológico, escribe una reflexión fascinante, pero hay dos cosas que uno tiene que tomar en cuenta cuando lees el libro. En primer lugar, como ha escrito Michel Morin, que ha analizado con mucho cuidado la obra de Monod, cuando uno lo lee, uno se da cuenta que los biólogos moleculares de la época creían estar descubriendo el secreto de la vida. Y el secreto de la vida parecía ser extraordinariamente complejo. DNA, proteínas, RNA, regulación. No se olviden que este libro lo escribe después del modelo del Operón, y le costaba mucho trabajo a Monod aceptar, como le costó mucho trabajo a esa generación de biólogos moleculares, que por puro azar esas estructuras y esos procesos pudieran aparecer. Pero como escribió Ernest Mayr, Monod era brillante, pero nunca entendió del todo el concepto de la biología molecular, de la selección natural, y nunca lo llevó a nivel subcelular. De manera que él estaba tratando de mostrar con toda justicia la debilidad de los conceptos basados en puras mutaciones, en puro azar, para tratar de explicar estructuras como la regulación de los genes. Cuando uno piensa en la evolución a nivel subcelular, tomando en cuenta procesos de transporte horizontal, de selección natural que opera sobre sistemas químicos, entonces la perspectiva cambia por completo. El otro punto que la gente ha omitido cuando lee a Monod, y no es trivial, es que Monod estaba involucrado en un debate muy fuerte con los antiguos miembros del Partido Comunista Francés, que eran de una rigidez ideológica extraordinaria, que habían apoyado a Lysenko, y el gran debate en realidad en ese momento entre muchos intelectuales y científicos franceses eran entre Monod y Pierre Grasset, que era de un estalinismo absolutamente radical. El libro de Monod hay que leerlo también como una crítica a la visión teleológica que tenían muchos marxistas de la época, y que era especialmente visible en el área de la biología, de la evolución. Monod fue miembro del Partido Comunista Francés, pero tuvo la inteligencia y la sensibilidad para renunciar muy rápidamente al mismo, ¿no? Y cuando fue necesario, estuvo del lado de ellos en contra de los nazis, ciertamente, ¿no? Al igual que François Jacob y que muchos más. Pero esos dos son factores que uno tiene que tomar en cuenta cuando lee a Monod. Ahora, Oparin en 1966 escribe un libro que se llama La vida, su naturaleza, su esencia y su evolución, en donde critica muy fuertemente a Monod. Cuando uno lee a Oparin se da uno cuenta que Oparin no leyó bien a Monod, igual que Monod tampoco leyó bien a Oparin.
Bueno, nada más un par de preguntas más para continuar.
Buenas tardes, doctor Lascano. Yo le quería preguntar lo siguiente. Usted nos dice que el propósito por el que hace usted toda esta semana de conferencias sobre el RNA es porque a los biólogos, cuando nos enseñan lo que es la biología molecular, nos está faltando incluirle la visión evolutiva en esto, a la biología molecular. Muchas veces en la carrera de biología nuestros maestros de biología molecular o genética son gente cercanas a otras áreas que no tienen que ver nada a veces con la evolución, por ejemplo, a áreas más cercanas a veces con cuestiones de biomedicina, de cosas así. Y pareciera a veces que la evolución es una cosa que a los estudiantes de biología curiosamente pues les entra de un lado y les sale del otro y lo abandonan inmediatamente, y otras áreas. ¿Usted cree que realmente a los biólogos a veces nos falta más incluirnos en nuestra propia formación la biología evolutiva?
Bueno, primero que nada déjeme decirle que el simposio no lo estoy organizando yo, sino lo está organizando el Colegio Nacional con el concurso por tripartito del doctor Bolívar, el doctor Juaristi y yo mismo, porque reconocemos la importancia que la biología del RNA tiene en nuestros días, y porque es una misión del Colegio Nacional divulgar ideas que se generan en otras partes y que tenemos que conocer y discutir con toda precisión y con toda amplitud. En segundo lugar, sí dije que uno de los objetivos, pero solo uno, era promover la enseñanza de la biología del RNA, porque es una excelente excusa para ver la evolución molecular a nivel de un cierto tipo de moléculas. Y en tercer lugar, yo creo que cada vez más, sí, cada vez más la biología en México se enseña, se divulga, se aprende, se practica empujados por una óptica evolutiva. Las razones son muy complejas, las podemos discutir en otra ocasión, pero yo sí creo que la primera ventaja que tenemos es que, a diferencia de lo que pasa en otras naciones, en México no hay un rechazo a la biología evolutiva. No hay un rechazo ideológico, no hay un rechazo religioso, no hay un rechazo social. No hay un rechazo como el que se da en Estados Unidos hace unos años, y no voy a mencionar a ningún político, sino a la entrevista que le hicieron a las candidatas a Miss USA para los concursos de Miss Universo, y les preguntaron qué opinaban de la evolución, y había 12 muchachas espléndidas, guapísimas, con una enorme vitalidad, productos muy acabados de la evolución, si lo quieren ver así, y ni una sola supo decir ni lo que era Darwin ni lo que era la selección natural, y de hecho la mayoría se manifestó en contra de la teoría de la enseñanza y la teoría de la evolución. Podría yo mencionar al presidente Pence, vicepresidente Pence en Estados Unidos, pero prefiero referirme a las concursantes para Miss USA. Eso no sucede en México. Eso no sucede en México. La gente aprende la biología evolutiva en la primaria, la refina en la secundaria, se enseña en la preparatoria, en el bachillerato, y yo creo que lo que tenemos que reconocer es la necesidad de desarrollar, refinar más aún esa enseñanza. Sin biología evolutiva en su sentido más amplio, no en el sentido de simplemente mutaciones y selección natural, sin esa visión de la biología que nos lleva automáticamente a interaccionar con químicos, con físicos, con médicos, etcétera, yo creo que estaríamos enseñando una biología que no tendría futuro.
Bueno, pues yo creo que aquí terminamos las preguntas. De todas maneras, vamos a tener toda la semana para que tengan acceso aquí a nuestros ponentes. Entonces, pues le damos un fuerte aplauso al doctor Lascano por su tiempo.
Gracias.
A continuación, vamos a tener el gusto de oír al doctor Ram Krishnamurti. Él es profesor asociado de Química del Departamento de Química del Scripps Research Institute en La Joya, California. Él obtuvo su doctorado en Química en la Universidad de Ohio, en Estados Unidos, en 1992, y desde esa época ha participado activamente en problemas asociados a la evolución química y el origen de la vida, siendo una referencia obligada para las investigaciones relacionadas a la síntesis de moléculas de importancia prebiótica. Desde el punto de vista químico, podríamos decir que la síntesis que el doctor Ram hace son una obra de arte químico. Al terminar sus estudios doctorales, ocupó una posición postdoctoral con el reconocido científico Albert Eschenmoser en el Instituto Tecnológico Suizo, ETH, en Zúrich. También ha colaborado con varias instituciones e involucrado en muchos proyectos importantes, como en del Centro para la Evolución Química de la NASA-NSF y en la colaboración Simposio sobre el origen de la vida, a la cual pertenecen premios Nobel. Coordina un grupo de investigación para el estudio del origen de la vida, en el cual en sus investigaciones utiliza las herramientas de la química orgánica sintética y metodología para entender, comprender los procesos químicos de la vida, sus orígenes y su evolución. En forma experimental, aborda cuestiones relativas a los orígenes de la vida con respecto a los monómeros funcionales y la aparición de polímeros informacionales. En su grupo de trabajo tienen el estudio de polímeros informáticos para ampliar la interfase entre ácidos nucleicos sintéticos y los biopolímeros naturales como las proteínas y los ácidos nucleicos. Propone nuevas herramientas moleculares que pueden proporcionar nuevas vías para sondear sistemas biológicos y para la investigación en lo que se conoce como biología sintética. Ha sido miembro de numerosos comités científicos y organización de eventos como las conferencias de ISOL, que va a ocurrir este año, Ram ha sido un visitante asiduo a nuestro país y hace algunos años participó en este mismo recinto en una serie de conferencias organizadas por el profesor Fioristi y por el profesor Guillermo Delgado. Por lo anterior, me es muy grato tener nuevamente a Ram esta tarde para que nos hable de sus investigaciones asociadas al mundo del RNA. Pues muchas gracias.
First of all, thank you to the organizers, personal invitation from Antonio to be here. It's wonderful to be back among friends again. So my talk is titled, Why versus How? And I'm not here to argue against or for an RNA world, but just to present you the difficulties of getting to any informational system in a pristine fashion, starting from prebiotic chemistry. That's the message that I hope to send through this. So this is taken from Jerry Joyce's Nature paper, where this is the approximate timelines in terms of the Earth's formation, stable hydrosphere, chemicals could accumulate and be stable, some chemistry happens, there is a pre-RNA world, which Antonio didn't talk about, and there is an RNA world, and then there is a first DNA, RNA protein world, and this is just an idea, hypothesis that people work with. And our interest lies in what happens in this area, this gray area of this prebiotic chemistry, pre-prebiotic chemistry to this pre-RNA world. Now, before that, I would like to start with a problem, and I assure you this problem has connection with science. Okay. So, father has 17 camels and 3 sons, and when father passed away, the sons open up the will, and this is the will. Half of the 17 should go to the first son, 1/3 of the 17 should go to this middle, and 1/9 of the 17 should go to the youngest. And you notice that there's a problem with the answer. You can't do this math and have a full camel at the end. So the question is how to solve the problem. And this is exactly the same problem we face with prebiotic chemistry to biotic chemistry. Now, there are 3 pillars of prebiotic chemistry. One is the foremost reaction, taking formaldehyde to sugars. The other is the very famous Stanley Miller's experiment, which takes simple gases and water to amino acids, also known as the Strecker reaction, and Oro's formation of adenine, where he takes simple molecules and makes adenine with just a self-assembly of these molecules. And it's very tempting to think, look, these simple molecules give biologically relevant molecules. Origin of life must be easy. Not only that, we also find these in experiments, in meteorites, and you find these kind of news-grabbing headlines, which says, you have these in meteorites. And now you put these experiments, which I showed you before, and these kind of headline-grabbing things, then you come up with solution that it must happen, because chemistry, prebiotic chemistry, astrochemistry is giving you the building blocks of life, right? But that's the prebiotic problem, which was also pointed out by you, that you have these things. These are nothing but the polymers of that. Why can't we go there? It's just a simple removal of water. But that's the black box that we are all facing. And I would argue that nobody has found a good solution to this, irrespective of any headline-grabbing news. Because the prebiotic chemistry that goes on here is incompatible. That means you take these molecules and put them back under the prebiotic conditions, they are not going to survive. Nor you take this and you do not get these things. And so the question is, how to solve the problem with what is given? Those are your 17 camels, and there is the will. And I'll come back to the will again. Is it the will of God or the will of man? Okay? So, this is the problem. Meteorites, John Oro's chemistry, Stanley Miller's chemistry will give you this. Formaldehyde will give you this. But you cannot put them together under any conditions so far. Okay? This has simply not been accomplished. So, prebiotic presence does not translate or guarantee further incorporation. None of these bases are reactive. So why would you want to form them? even if you get them in meteorites, they are not going to react. So what is the use of those? They are completely insoluble, unreactive. So nothing is going to happen. Yes, it's so nice to see them in meteorites and in life. But the question is, are they useful so early in prebiotic chemistry? Structural proximity, because look how close they are in structure, does not mean prebiotic reactivity. That's a point that you need to really keep in mind, because there can be misleading clues that will take you in so many directions that you will be unproductive. But if you allow for alternative reactants that are not here, which are not biologically relevant, but may be prebiotically relevant, and not start from the fully formed biological molecules, you can get some answers. And this has been shown. Leslie Orgel very famously showed that if you use these kind of molecules, which are not in biotic chemistry, but in prebiotic chemistry, you can get to these kind of molecules and nucleotides and nucleosides. Nagware showed long time ago that you can convert arabinose to ribose. So why do you want to go look for ribose from the very beginning when another sugar can also give you this answer? So you have to take into account what all might have been there rather than just focusing only on what life is using today. It's a current theme that I'll come back to every time. Now, John Sutherland and Powner did a beautiful job of putting all this together. And they showed that you can start from even simpler, potentially prebiotic molecules. Look, urea, cyanamide, glycolaldehyde, and glyceraldehyde. But the kicker is, but none of these are biologically relevant today. Now, nature does not use it. But you still leads to the biologically relevant nucleotides. So that means to look for biology in prebiotic chemistry might be a misleading way to think about solving the origins of life problem. Now, more recently, there has been the Thomas Carell work where they showed even simpler systems, and they synthesized the purine nucleobases and nucleosides, which John and Powner could not. So you have this very nice demonstration starting from molecules which have no connection to biology, but end up in molecules that could be used in biology. Now, so this is the way. So the formation of RNA, the question is, how is RNA formed? That's the question that people are asking in that sort of experiments. How is RNA formed? What are the pathways with which I can build RNA molecules, nucleotides, nucleosides? Now, so they start with a slew of potentially prebiotic inventory, and they narrow it down to RNA-specific precursors. They don't want anything else. They don't want anything that would interfere. They want RNA-specific precursors. They want RNA building blocks and RNA only. It's the RNA only way. That means we want RNA from prebiotic chemistry. Nothing else. Others, I will just push it aside. If there is a side reaction, I will formulate an experiment which will overcome the side reaction. I will look for conditions which will make the side reaction go away, but only focus on RNA. Now, this I'm repeating many times, because if this was something that we would object to, okay? This is, as scientists, we would object to, right? We would also object to this, right? Now, then we must also object to this. RNA is a destination, not a destiny. That means to focus only on RNA molecules from the very beginning is probably not the best way to convince the general public that, there is a different way of how life originated. Because we are playing the same role. We are becoming the creators of RNA, if you will, by focusing only on RNA. So, now come to the Eschenmoser approach. And I'm really glad that I joined his group and got trained by him. The question is why? The question is, why RNA? Why not something else? Life uses so many other sugars, like glucose and fructose. In fact, you can do this test. Just walk to anybody and say, give me a name of a sugar. And they'll give you glucose, fructose, but rarely will they say ribose, because ribose is not considered to be a sugar, because it's part of an RNA. So that's the general public perception. So the question is, you look at that, and it has a lot of parts to it. And I'm glad that Antonio gave a good introduction to that. And there is these two difficult bonds that are to be formed in water. The question is, you can ask this question, why a pentose? Why this 5-carbon? Why then, if it's a 5-carbon, why a ribose? Why a ribofuranose? Why not a ribopyranose? Why a phosphate? And why these 4 bases? What is so special about them? I told you that they are not reactive. Then how did nature choose such non-reactive bases to be part of this most important molecule of life? So why RNA? And if you keep on asking this why, you come to a conclusion that it's a highly optimized structure. And I will try to convince you by just giving only one example. There are many of why I think this is a highly optimized molecule. Let's go to and so the question of why, was beautifully, enunciated by Eschenmoser in this very nice paper. I urge people to read this again and again because it's every time you read it, you get a different message. So there is the RNA, and there is not only ribose in many of the prebiotic reactions, but there are so many other sugars. How did nature decide to select that ribose? At what level did it select the ribose? Did it select it in prebiotic chemistry, or did it select it at a higher level? There is also the uloses. These are the corresponding keto version of the ribose and the keto version of the xylolixyl. And these are actually formed in greater extent in prebiotic reactions such as the foremost, not the ribose. The pentuloses are one of the major components. Why did nature not choose those if that prebiotic chemistry pathway is the way to lead to sugars? There are different bases which do base pair, and they are also potentially prebiotic. In fact, there's a beautiful paper by Miller who did an experiment unknowingly, not willingly, for 27 years at minus 78 degrees. They took NH4C and ammonium cyanide, forgot about it, and when he was retiring, they cleaned out the lab, and ammonium cyanide gave a slew of bases. Not only just adenine, guanine, cytosine, thymine, it also gave a lot of these bases. So why did nature not choose those? We choose to focus on what nature is using now, and that is, I want to focus on that. And that is a problem. There are different backbones. These are glyceric acid backbones, and glyceric acid is another one. There are, these are pentuloses. There are different types of sugar backbones that function. They also have a lot of things. And the question is, why this guy, right in the middle, chosen by life? And again, we come back to this conclusion, it's an optimized structure. So what I will do next is try to show you and convince you that it is so, by just focusing on the bases. This is the canonical Watson-Crick base pairing. The uracil goes with adenine, cytosine goes with guanine, and it's a purine-pyrimidine. And the question is, why? Why can't you have purine-purine or pyrimidine-pyrimidine? And it has been argued by Leslie Orgel, no less than Leslie Orgel, that purine-purine might have been the forerunners of the purine-pyrimidine. So why not these? So let's go through a little bit of excursion. You know this. These are phospholipids. And this is a phenomenon encountered by everybody during the time they take bath. You make these kind of wonderful phospholipids, vesicles, which take out your dirt and wash it away when you have a bath. And they do concentrate and self-organize, and they give this beautiful phospholipid bilayer where you have the charges, the phosphodiester charges facing the water and the hydrophobic core inside. And the thing is, if you also look at nucleic acids this way, you can think of them as a polynucleotide bilayer where you have a hydrophilic exterior, a hydrophobic interior, and these are, because the hydrophobic forces push them together. Now, how does this help in understanding the structure of RNA and the bases? So there is a hydrophobic-hydrophilic balance which actually makes the purine-pyrimidine the best paradigm. And it's as follows. So the longer bars are purines, the shorter ones are pyrimidines. And there you have it. And the phosphodiesters are outside, and you have an electrostatic repulsion between the two phosphodiesters, which is balanced by the hydrophobic nature of the bases inside. Let's go to purine-purine. And you see what has happened. You have increased this distance. You have made the hydrophobic character greater, which lends greater stability to the duplex. You have made the phosphodiester's go far away, decrease the electrostatic repulsion, which means that duplex is much more stable than this guy, the purine-pyrimidine. You go to a pyrimidine, you do the opposite. You compromise the duplex stability because the phosphodiester's are close, the hydrophobic interaction is slow. So one would argue that probably this is the best for base pairing. Now, this is a hypothesis, of course, but luckily, there are some indications in the literature that this may be right. The work of Eric Kuhl, who worked with extended bases, what he did was instead of having cytosine or uracil, he extended the uracil, extended the cytosine. So what you have are purine bases here. And what happens when he does the base pairing and studies the mismatch discrimination? There it is. This extended base pairs, they still tolerate mismatches because they are much stronger. Of course, when you like a person much better than another one, you will tolerate whatever the mistake they make. But if there is a person that you don't like, even the slightest, you think they may have made a mistake, they are gone from your life. Okay? The same way, DNA, because of this purine-pyrimidine, is so sensitive, and that is what you would want in an informational system. You don't want to have mismatch, tolerance to mismatch discrimination. And this purine-pyrimidine manifesto does not happen at the level of a monomer. Just because I made adenine, just because I made adenosine, you don't get this purine-pyrimidine selection. You get the purine-pyrimidine selection only at the level of an oligomer, not at the level of monomer. So to claim, I synthesize purine nucleotides, I synthesize pyrimidine nucleotides, therefore RNA, does not quite cut it. You have to come to a level of an oligomer at an emergent property level to get the purine-pyrimidine working. So that means it's an optimization, not the maximization of base pairing strength within the context, and it's important, of a ribofuranosyl phosphate backbone. Because if you change the sugars, the rules change again. You will end up with a purine-purine or a pyrimidine-pyrimidine, depending on what that system would prefer based on its backbone. So all these things that you are seeing at the level of RNA, beautiful properties that Antonio was talking about, is at the level of an oligomer, not at the level of a monomer. So prebiotic chemistry delivers you the monomers, okay? Not yet capable of delivering the oligomers. But even if it does, that property of a purine-pyrimidine selection comes at a much higher level, not at the monomer level. So just to summarize, the ribofuranose optimizes, and you have to have the aldose form of the ribose. It's in a furanosyl form. The nucleobases are the beta position, and it's the 3' 5', not the 2' 5' linkage. The pKa of the nucleobases play an important role. They must be in the keto form, not in the enol form. And it's, again, an optimization process. Pyrimidine-purine selection, Watson-Crick selection is again at the level of an oligomer, not at the monomer. And RNA's attributes are fully manifested in only an aqueous medium. You change the medium, you change the pKa of the medium, RNA's function goes away. It's very important. You know, people forget the environment. They talk about RNA, they talk about proteins, they talk about this. But the environment is so important. The pH, you look at extremophiles who live at pH 11 or pH 12 or pH 1 or pH 2. They do this fantastic chemistry of keeping the protons in or keeping the protons out using proton pumps to maintain a cell pH of around neutrality of 6 to 7 so that your base pair can function, your protein can function. So to separate the molecules from their environment would also be another issue for origins of life. So the chemistry and biology of RNA indicates it's a highly optimized molecule for a given environment. So imagine the prebiotic chemistry environment 4 billion years ago was totally different. And people still argue about what is this, what is it, what is it. Irrespective of that, we must be cognizant of that. So my take is that RNA is likely a product of chemical evolution. Remember, this does not argue for or against an RNA world. It's only talking about how RNA might have emerged as a molecule. So going back to this, selection of components at RNA at what level? You can select here, which many prebiotic chemists tend to do because it makes the problem easier. I make only ribonucleotides, RNA. Then there are people who say, oh no, I can do the selection here, or here, or here. And you see, there are different criteria. Here, this is soluble in water, completely water insoluble. You mix them both, enhance solubility in water, stability of the nucleobases. Now, if you go here, there is ability to form polymers with recognition capabilities. Here, hydrogen bonding in water. And remember, that is one other problem that I didn't bring up. The monomers do not form hydrogen bonds in water. You need to come up to a 6-mer or an 8-mer level before they can start doing it. So those are the problems. That means there are opportunities at each level with increasing demands and selection as you go from the elementary to the more supramolecular level. And to start saying, I need all the selection here, would be kind of defeating yourself in what you want to demonstrate. For example, this is ribulose. It can still form the nucleoside because it has the same anomeric position. And it can still phosphorate, but it will fail at this because we know that we made this oligomer, it does not base pair. So that means I can look for ribose and ribulose being together, but allow for a selection at a much higher level and not want RNA immediately at the very beginning. So what about this emergence of RNA? You have, again, the same thing, and there was the RNA only way that I talked about. And there are people who want this pre-RNA because they say, oh, RNA is completely unstable, therefore there must be pre-RNA which is more prebiotically feasible, and maybe it gave rise to RNA. And I have problems with that as well. So I will point that out. So the question is, with a fully homogeneous pre-RNA candidate, now how does this, they wanted to look and act like RNA. How much of RNA character can you force on this pre-RNA? Okay? Is base pairing to RNA an absolute requirement? I'll come to that. You will think it is almost a given. It must base pair. Or else how would it transfer information? I'll come to that. How much information and functionality do these primitive pre-RNA candidates, should they evolve, or should they just give rise to only RNA? How does this pre-RNA candidate invent the chemistries to give rise to RNA? What would it have to do? And how do you transfer the function of this pre-RNA to an RNA? Or does RNA have to get it de novo completely? So we are going to play a mind game, a Gedanken experiment. It's only a thought experiment. Don't take it seriously. Don't argue with me about this. It's only a thought experiment. So you have RNA and DNA is supposed to have come after RNA. Okay? And therefore, you would say, is base pairing between these two necessary? And you'll say, absolutely. Otherwise, we will not be alive. Now let's go to a pre-RNA. If you do believe in a pre-RNA, and you ask, is base pairing with RNA and DNA absolutely necessary? And you'll say, oh, probably with RNA, not DNA, because DNA came later. Okay? So we allow for that. Then we go to a pre-RNA. Okay? And then ask the same question. Remember, this is a thought experiment. And then you say, yes, but it's only with the pre-RNA. And then you go to a pre-RNA. And then you say, what should I do? And you say, this, but then this and RNA need not talk. They are so far apart. They may be two different species. So to find a primordial informational system and to claim that it doesn't base pair with RNA, therefore it isn't, may not also cut it because you may be too primitive and too far away from RNA. And if you don't believe me about this, just think of your computer systems. Okay? Now, this is a slide that I made a few years ago, so I have not gone past OS line, but try taking a file that was made here and put it there, or try taking a file that was made here and put it here. It would not talk to each other. They are two different operating systems, highly evolved operating systems that cannot talk to each other anymore. Okay, and remember, this is just to make the point, there is also problems with the pre-RNA world. That means you have to account for how you would move from a pre-RNA to an RNA. Okay, base pairing with RNA, and so that would also be no. So I have problems with both the RNA first or the pre-RNA first approaches. And what are those? The chemistries, if you look realistic prebiotic chemistry, they are messy. If there is any synthetic organic chemist in the audience, they will know how side reactions can kill your project. Okay? Reactions are not very clean. If you want it to be clean, you need to kind of interfere. Prebiotic clutter is the rule rather than the exception. And prebiotic chemistry is really inherently messy. And you see these meteorites and these wonderful headlines, nucleobases found. What about the remaining 99.9999% of things that they found were not reported because they are not used by biology? Okay? So one has to be extremely careful about extrapolating such things. There is what is known as systems chemistry that is coming up, and selection is the key. How do you select from messy stuff? And then, as I pointed out, selections can be at the level of different, the prebiotic level, the oligomer level, the polymer level, and you should allow for all levels of selection to get to your system that would be the one that is functional. So, what sort of things that would be happening here? It's not only ribose, but all other possibilities, all other linkers, all other nucleobases. And would they select themselves out based on selective reactions, form selective bonds, and lead to this, which may end up in functional selection there? And selection for RNA, only for RNA at the monomer level, is kind of defeating the purpose, as I said before, but there is selection based on many different levels. And this goes to the question of teleology that was raised before. There is no teleology in prebiotic chemistry. When you do a prebiotic chemistry, it doesn't know it wants RNA. You direct it towards RNA by shining light at the appropriate moment, cutting off light at the appropriate moment, adding this molecule at the appropriate moment, removing the molecule at the appropriate moment. That is not prebiotic chemistry. That is interventionist chemistry. There is no teleology, a purposeful destination such as RNA in prebiotic chemistry. And that is what I want to emphasize, not arguing against the RNA world. So, can you have a systems chemistry towards the emergence of RNA? What is the paradigm that nature uses? It always uses heterogeneity in the nucleobases, but homogeneity in the backbone. Can we now change it based on the prebiotic clutter that I have talked about? Can you have heterogeneity in the backbone and heterogeneity in the recognition element and still move towards a homogeneous backbone system? Is that possible? Or should we always think of homogeneous system to a homogeneous system? And this goes to the question of RNA DNA. Now, the RNA world theory, which holds that RNA invented proteins, proteins invented DNA. Remember, they didn't say RNA invented DNA, because so far there is no ribozyme that can do that. So they always conveniently take it from biology and say RNA invented proteins, proteins invented DNA. And therefore the problem is solved. And that is a point that I'll come back to, that the RNA world theory, if you look very closely and in the literature, is an extrapolation of what extant biology does, and it's not a product of prebiotic chemistry thinking. It's always coming from pushing extant biology back as far as possible, getting help from prebiotic chemistry when needed for RNA. That's it. So, how does RNA go to DNA? Okay? Now, you know what biology does, but how did it happen in an RNA world? Let me call it pre-DNA and put the same impositions that we put on a pre-RNA on pre-DNA and ask the same questions. We should be able to, right? We are scientists. So, our RNA world is based on inferences obtained from biology than prebiotic chemistry. It's important to note that. How far can you extrapolate the observation based on extant biology into the realm of prebiological chemistry? Now, this is the same question that I asked about RNA and pre-RNA. How far can I push the requirements of RNA into pre-RNA? Okay, it's a fair question. How does RNA world give rise to DNA? How does RNA world invent the chemistries necessary for DNA precursors? Proteins, of course. That again is an extrapolation of biology. How are the chemistries of RNA and DNA nucleosides and nucleotides kept separate to avoid heterogeneity? Now, we know biology goes to a great extent to keep RNA and DNA separate. Even if there is a slight mix, an enzyme comes, completely cleans it out. If a deoxycytosine deaminates to give you a deoxyuridine, there is an enzyme that fixes that and keeps everything separate. Now, in an RNA world, which would have done the job? There is no sophisticated enzymes when RNA was just inventing DNA to keep them separate. How did it do that? So, what we did was we just decided to have some fun. And remember, RNA pairs with DNA beautifully. A homogeneous DNA and a homogeneous RNA pair very well. What happens if you mix them? So what we decided to do was ask this question, can you have dRNA and rDNA and still make this transition? Can you go from a heterogeneous to a homogeneous transition? So the question was this. So what we did was we started with the AU8 of RNA and went to 88 by mixing in U and T's and A and A's of deoxy and ribo. And what we found was that here is pure RNA, and here is pure DNA. And whenever you started to mix them, the chimeric sequences were always weaker. There was nothing comparable of stability to RNA, pure RNA or pure DNA. Now, this is a self-complementary system. We also did it for self-complementary, non-self-complementary ATGC containing sequences, pure GC sequences, and without exception, it's only the pure homogeneous systems that base pair and the heterogeneous systems do not. So for you to go from a pure RNA to pure DNA, you would have to avoid this mixture because they may be lethal, because they don't base pair. They don't. So rather than this, and we also looked at the thermodynamics of this sequence for this particular one. And here is pure DNA, and these are the delta G values. And here is pure DNA. There is only one sequence that even comes close to the parent sequences. And interestingly, that is a combination of purines on the RNA and the pyrimidines on the DNA. You change that, you go to a highly unstable system. Okay? Now, to put it even in a more dramatic graphic fashion, here is pure RNA, pure DNA. Notice the black color for RNA, the red color for DNA. And if you start mixing them in any which way fashion, you always end up with systems that are always unstable thermodynamically and thermally compared to the parent systems. So the question then becomes, what do you do? So this RNA world, it faces the same questions, faces the same problem that a pre-RNA to RNA would face. A homogeneous RNA to a homogeneous DNA seems unrealistic. An RNA world would have to have some highly sophisticated mechanism to avoid this prebiotic clutter. And imagine, I have not even introduced the D and L problem, the chirality problem. You mix D sugars with L sugars, that's another story where it leads to completely destabilized duplexes. So chirality has not yet been touched here. RNA needs other important actors in order to co-evolve. That means an RNA-only world is not realistic, and it needs to be rethought. Imagine, all the experiments that you are going to hear today with RNA world, and tomorrow, and till Friday, are all done in test tubes. I don't think there was Sigma Aldrich 4 billion years ago to sell you test tubes, okay, to do these experiments. It must be done in more prebiotic. Expose RNA to a mineral test tube. It is going to decompose. And so the question is, what RNA needs? For example, this could have been in a vesicle. So then it's not an RNA world anymore by the definition of RNA only. It's an RNA in a lipid or a vesicle, which would have done the chemistry. Okay? So going from RNA to RNA, somehow through these highly unstable intermediates, rather than that, why not allow for RNA and DNA to form through prebiotic chemistry? Everybody wants only RNA by prebiotic chemistry. Why not DNA? That is because you have been forced to think that DNA comes after RNA by taking biology and extrapolating it. You don't want DNA prebiotically? Why would you not want it? It would make life simpler. Says not me, but the very people who want an RNA world. Sutherland, when our Angewandte Chemie paper appeared, he wrote me a short note saying, Ram, we also thought of this. I said, fine, that's good. And he says, look, a predisposed route to rDNA could be potentially even more significant. And this is the same Sutherland who published the 2009 paper that everybody wants to talk about as a support for an RNA world. Jack Szostak says DNA precursors in water. And look what he says. The primordial biochemical exploitation of a mixed RNA-DNA system could eliminate the requirement for a genetic takeover, the same problem of genetic takeover that I've been talking with the pre-RNA to RNA. Thus, RNA and DNA may have emerged as early contemporaneous specialization of a primitive mixed biopolymer. I think now people will start listening to me because the two great people have said so. So, can we go do this? Can we start with these mixed building blocks? RNA, pre-RNA, everything is there possible in prebiotic chemistry. Can we go to homogeneity by making these Occasionally, you make stretches of homogeneous strands. And what is known as homogeneous strands come together, make duplexes, and they are more stable to hydrolysis. Single strands hydrolyze back. So you can have a hydrolytic stability selection for structures that form tertiary structures or duplexes. And you could move to a pure homogeneous system. And the thing is, base pair mediated information transfer is not the sole criteria, but is an important criteria. Hydrolytic stability, interaction with other molecules, capacity to be encapsulated. There are so many things that are possible in prebiotic chemistry which we tend to overlook by focusing only on the RNA world. RNA and DNA could have been a product of incremental constitutional replacement, not necessarily a wholesale replacement of one backbone to another homogeneous backbone. And that would make perfect sense in a prebiotic world. So, can this be done? I will just show you hints of whether it is possible. Here is TNA, which is a 3-furanosyl backbone RNA. Now, if you take RNA and RNA, which is in black in color, they base pair very nicely. You take TNA, they also base pair very nicely. These are UVTM values, thermal values telling you how stable the duplex is. Bigger the number, stronger the duplex formation. And here is TNA homogeneous backbone with RNA homogeneous backbone. It also forms a good duplex. Now, what we decided to do was take a mixture of this. That means it is heterogeneous both in the backbone and in the nucleobases. So it is a mixture of backbones and mixture of bases. And we thought it should pair. It does not. We were crushed. Bam! It did not work. But then, out of the blue, I told Subhendu, who worked on this, see whether it base pairs with homogeneous backbones. It was a totally off-the-wall suggestion. We didn't expect anything. But look, these do base pair with the corresponding complementary homogeneous backbones. And what does that tell you? Immediately, people can think of experiments where heterogeneous backbone sequences can act as templates for replication of homogeneous backbone sequences. That means you can go from this blue-black mixture to a pure black or a pure blue backbone because of this capacity. I still remember Jack Sostak once asking me about the DNA RNA paper saying, but I don't understand the role of your heterogeneous backbones. What would they do? Now we have an answer. And we have preliminary data, which I will not show today, that this indeed can be done. That you can take these kind of heterogeneous backbones and move to homogeneous backbones by chemical ligation, by them acting as templates. Now, this is the slide I think everybody is looking for, right? You want an answer to that. Okay? Other things were not that important. So, you always go to a wise old man to solve problems, right? So, they went to a wise old man, and he brought a camel of his own. He added to the mix. And now he started reading the will, and math became quite easy. So he gave 9 to the eldest son, 6 to the middle, and 2 to the camels. And you know what is the most important thing? Add the numbers, it is 17. Okay? He takes his camel back. And that is exactly what we are doing. We are looking only at what biology wants and look into prebiotic chemistry, get it. And we don't get it, so we come up with fantastic schemes and claim prebioticity and support for something. And I will show you that this is possible, but not with RNA, but with peptides, which we have published. So this is what everybody wants, because this is produced in Miller-Urey experiment. This is in meteorites, okay? But many people have spent their life doing this without much success or using artificial activating agents, claiming its prebiotic relevance, etc., etc. Okay? Now, if you go a little bit away from the alpha amino acid to its cousin, which is called the alpha hydroxy acid, that polymerizes by simple heating. Polyesters are well known. Okay, remember, if you just replace the NH2 there, it's an alpha amino acid. These can do it, and people have done that. And organic chemistry teaches us if you make an ester bond, you can attack it with a nucleophile like an amine to get amides. And people like Alex Rich have said that, it is possible that early polymerizing mechanism may have resulted in polymers containing both amides and esters. And interestingly, this paper appeared in Nature, but did not get written up in the New York Times. You know why? Because it focused on alpha carbohydroxy acids. It didn't say alpha amino acids. If they had said amino acids, New York Times would have published a front page paper on it. But it was alpha hydroxy acid. Biology does not use them. What is the use? But it is in the meteorites, but nobody focuses on that. But you can, as a scientist, go and ask the question, what if I do a mixture of alpha hydroxy acids and alpha amino acids? Would it get me something? And this is the idea. You take these mixtures, and you know that this forms from organic chemistry. Then you do an exchange. This is an amino group which attacks the ester, makes an amide bond. Still has a free carboxylic acid. Go ahead and do another esterification. And you continue this, you continue this, you continue this. You can see by the simple method of mixing alpha hydroxy acids and alpha amino acids, only heating and drying, cooling and wetting, you can produce a deoxypeptide which has nothing but enriched peptide bonds in the middle. Does this work? Of course, or else why would I be presenting it here? So you take mixture of amino acids, and this is what you do. Just warm dry phase, heat at 85 degrees, cool night phase, add water and leave it, and then heat it. That's all. No fancy activating agents, no other chemicals. These are things that are available both in the Miller experiment and in the meteorites. Glycine alone, no surprise, does not work. This is a mass spec. Lactic acid alone, look, beautiful polymerization. Mix them together, you get deoxypeptides. Amino acids and amides, peptides and esters mixed together. And you can do the cycling. So this is cycle number 1, cycle number 5. And you see how the lactic acid vanishes and more amino acid peaks grow in. You can do this and follow it, and you can measure how many peptide bonds are there. You can also follow it by IR, and you can show how the IR bands for the amino acid peptide bond formation grows up. You can add more lactic acid because it's an open system, the lactic acid evaporates, you add more, you get more peptide bond formation. You can go in and sequence by MS-MS sequencing and show, yes, actually, according to what the mechanism predicts, you have nothing but peptide bonds in the middle. You can do it with alanine, you can do it a mixture of things, and you can prove that this is what you have. So, this is difficult under prebiotic conditions because you are focusing on what nature uses and what prebiotic chemistry gives you. But if you also allow for other actors in prebiotic chemistry, which are there, but which we tend to overlook, this is possible. That means by considering what other molecules may be present, the biologically relevant molecules may become possible and may become easier. And that's why I said I'm not arguing against the RNA world, but I'm arguing against how the RNA world emerges. Is it a Y RNA or a how RNA? And now I give you the same diagram that I showed you for RNA, and probably you will say, this is more believable because I have demonstrated it. And the question is that alpha hydroxy acid your 18th camel? And it is. And we tend to overlook that. So we don't really have to go to a wise old man for that alpha hydroxy acid. It's already available in the meteorites and Stanley Miller's experiment. So this is the central problem. What is demonstrated by prebiotic chemistry is not compatible with what extant biology is based on. We have to recognize it. To shut our eyes to that is kind of fooling ourselves. And what is used by biology is inconsistent with what prebiotic chemistry can assemble. Use the word assemble and not form. How do you bridge the gap? That's the answer. If you can believe that this guy evolved into this guy, all you have to do is allow the same freedom for molecules to evolve, and that's chemical evolution. And with that, I end the talk and give you this slide. I need to thank my co-workers who did all the work, and it was the ester-peptide exchange was a beautiful collaboration between the groups at Georgia Tech, between Nick Hart, Martha Grover, and Facundo Fernandez, and the funding agencies for making this possible. Once again, my heartfelt thanks to Antonio Lascano and the organizers for giving me this opportunity to present. and I'll be happy to answer any questions that you have. Thank you.
Muchísimas gracias por esta conferencia tan interesante donde vimos la anatomía de una molécula. La conferencia está abierta para preguntas.
I hope I didn't scare people off.
Ram, that was a beautiful talk. You seem to be focusing exclusively on duplex formation when you talk. Yeah, I'm speaking English. You don't need to translate me. You seem to be focusing exclusively on duplex formation when you talk about the selection for RNA and the transitions. But if you look at real RNA, stem loops and all these other structures had to be very important. I mean, could you just comment on that a little bit?
I'll move over to the podium again because you are absolutely right. I only mentioned it very cursorily in my colorful slides of moving from mixtures to that, interaction with other molecules forming tertiary structures. Duplex, again, it's a kind of a hold on people to focus on duplex stability, but you're absolutely right to focus on the catalytic property. In fact, I remember having an argument with Nick, and I think you agreed more with me than with Nick, saying that it's probably the catalytic capabilities that probably drove things first rather than duplex formation. And I would agree with that, which probably also goes for Michael Yaros's work, where he shows beautiful catalytic activity of single-stranded RNAs.
Sorry, I still have a question about one of the precursor. I don't have to.
Yeah, can you stand up a little, or else I'm not able to.
Sorry, I'm not an English speaker, but I will try. I still have a question about one of the precursors. I don't know how to say it. Of the how that this ribose could be phosphatated if.
How come ribose can be phosphorylated?
Yeah, it is a precursor of the RNA. So I have a question about if is there a theory about how it was created?
Okay, so if I understand your question correctly, it is how ribose gets phosphorylated.
Oh, no, but
maybe you can talk in Spanish and they would translate.
Yeah.
Yeah.
Tengo una pregunta sobre cómo es que fue creado uno de estos precursores del RNA, como el expositor anterior estaba diciendo. Bueno, lo que yo sé es que esta fosfatación debe de ser con una enzima o una enzima. Entonces, este RNA se supone que ese es un precursor, es el precursor de las coenzimas. Entonces, pues, quisiera preguntarle si hay alguna teoría que diga cómo es que este precursor fue creado, pues, antes del RNA, si no había coenzimas, no había enzimas.
Yeah, I understand. Okay. So, the question basically is, how does the phosphorylation process happen to give you these nucleotides which form the backbone of RNA, if I understand it correctly. So there are many experiments that have been carried out by pioneers such as Leslie Orgel and others who have shown that you can use phosphates available in minerals, phosphates available as various forms of phosphates such as struvite, which is a magnesium mixture of phosphate. And there are people who have used alternative solvents apart from water, such as formamide, to get the phosphorylation growing. The only problem with all such experiments is that you have to use a completely different environment to do the phosphorylation, and then you have to take them into water to do the elongation. And so there is a problem with that. There are solutions which I cannot talk about right now because these are under consideration for publication, that if you think, just as I told you, you are only focusing on phosphate, what nature uses, right? But if you move a little bit away and think of nitrogenous versions of the phosphate, you can actually come up with phosphorylating reagents which will do this reaction beautifully in water and not only stop at phosphorylating, but I also can give you oligonucleotides if you want to. So that means there are solutions that have been pioneered by great people before, but that has its drawbacks because you have to move phosphorylation in one system and then move to a completely different system where you can do the elongation, and then to a completely different system where it will be functional. To do it in a one-pot scenario in water would be very pleasing intellectually and would overcome a lot of the arguments people have against a prebiotic chemistry demonstration of that.
¿Otra pregunta?
Thank you for your talk. I can recall that in a book of Degens, he said he was a very versatile researcher, as I recall. And in his book, he says that, or he said that by knowing the molar values of the calcium inside the cells, it was possible to know the possible values of the ion strength in the ancient Earth.
Yeah.
So you said that that knowing the features of your self-assemblies, let's say the distance between the both chains, is it possible to know some of the ancient features of the Earth, of the ancient Earth, by knowing what's let's say the optimized structures?
Yeah.
Let's say like the distances, if you are using different temperatures or different concentrations of ions.
It may be possible, but again, to take what is present today can only provide you clues. And the problem with clues is the clues can give you a good answer, but they can also be misleading. Mm-hmm. Because of the uncertainty of what existed 4 billion years ago or 3.8 billion years ago. There is some fair idea about what the atmosphere might have been, what would have been the ocean composition, what is the partial pressures of CO2, and things like that. But still, there is not a well-settled evidence that would conclusively say, this is what you should be looking at. For example, many people want the origins of life in the ocean, which is almost impossible because of the dilution problem and the instability of the biological molecules. So people now favor smaller pores or smaller containers where this chemistry could have happened. So they are going into niches. And the question is, can we extrapolate those concentrations that we have today to the ocean or to the niches? And that is where the problem comes.
Possible you don't need even an ocean, just mineral surfaces.
Yes.
Or vessels, something like this.
Yes.
Thank you.
Thank you very much for your lecture. And my question goes in the sense, which is the status of the catalytic, or the catalytic status of this pre-RNA world, taking into account the hydroxy acids and the heterogeneous nucleic acids? Because in the traditional, let's say, RNA world, you have like a very nice way of solve some catalytic reactions. But in this pre-RNA world, how can that be done? Thank you.
Very good question. Very good question. And this goes to one of the problems of how do they have to have catalytic activity, and do they have to transfer this catalytic activity to RNA? The answer is probably not. They can have different catalytic activity, and RNA must de novo, because the structure is so different. It may have some memory, but it may not. Now, people have worked with different backbones. And Jean Chaput has shown that if you work with the triofuranosal backbone, which I showed, you can create triozymes. That means a different sugar backbone with the same 4 canonical nucleobases can make enzymes. And now there are people who are pushing whether ribozymes can make different sugar backbone oligomers as well. So people are slowly beginning to open up to the idea that there might have been mixtures. And now people are even beginning to look at arabinos, thanks to the work by Donna Blackman, which appeared in ACS Central Science, who showed if you want to hydrolyze aminonitrile, you use a ribose, D-ribose, you get the wrong amino acid chirality. You use L-arabinos, you get the right chirality. So if you wanted ribose from the very beginning, it's going to give you the wrong amino acid chirality, according to what her experiment shows. So now people are focusing on arabinos. So it's good to focus on what library of compounds would have naturally given out, rather than actually going in and saying, this is the pathway I would take because that is where we are right now.
Thank you.
Oh. Hello, Ram. Nice to meet you again. Very beautiful talk. I always like it. I have a question. You are no longer constrained by the pairing with the RNA that was before, I remember. So the pre-RNA should be focused not only as simple structure that can be assembled easily, but also they must be more stable in a geological scale of time, something like that? Pair stronger maybe, or not?
So the base pairing strength is something that, which is a moving target. As I pointed out, strong base pairing does not mean optimal base pairing for that particular function. If you want a function within that particular environment, the structures would have to adjust to that environment to give that function based on the tertiary structure that they would form. So if you take RNA, which is catalytic at, say, at pH 6, and force it down to pH 3 or pH 9 or 11, it's not going to be catalytic, even though there was that base pairing at pH 7. So it's a very moving target, if you will, and this would be true for the pre-RNA as well. So what is true for the RNA, that argument will hold good also for the pre-RNA. So it would be very difficult for me to give a precise answer to that, because we are still exploring what is around RNA, which could have given rise to that.
A beautiful talk. I'm going to ask you to do the same philosophical exercise that you're criticizing about the RNA biology theory. Do you know if there's any footprint of that complexity in chemistry that is still extant today in modern biology?
I won't say
is there anything that could bring you back to that?
I won't say complexity, but I can point to one molecule that is a favorite of mine, orotic acid. Now, if you look at the de novo pyrimidine synthesis, nature does not take uracil, preformed uracil, and put it with ribose. It takes preformed orotic acid, makes orotidine. Now, the uracil that it combines with ribosyl 5-phosphate, or the diphosphate, 1' diphosphate, is the one from the salvage pathway. So you don't see biology synthesizing all the 4 canonical nucleobases de novo. It always comes from orotidine. But the kicker is, orotidine is not present in RNA or DNA in the informational system. So what is orotidine doing there? And orotidine is produced 50 times more in the ammonium cyanide experiment that I showed of Stanley Miller, I talked about, than uracil. So why not consider orotic acid? People have never focused on nucleosidation of orotic acid because orotic acid is not in RNA. And that is exactly what I'm saying. So there might be some clues, but I'm not sure whether the clues are there to lead or mislead. Tony, go easy on me.
No, actually, going precisely to the case of orotic acid, I don't know if he ever published it, but I remember Jim Ferris saying many times that orotic acid would not incorporate easily into RNA because it decarboxylates so easily.
It decarboxylates as you can decarboxylate by
Right. And there is a slight mention of that in that chapter that he and Jeff Subay wrote for the biochemistry book. But if I understand you properly, and I love what you say, I was strongly reminded of a long-standing argument that we have when working with biology, that you cannot extrapolate phylogenetic trees back to the waters of the primitive ocean. So what you are really saying is that we shouldn't extrapolate the further back we can go from biology is to RNA, enzymes, coenzymes, whatever, and what happened before is a very shadowy area in which we need to work together with chemists and structural chemistry, plus metals, which are basically absent in the discussion that you made.
Yeah. So I would say, the same analogy can be drawn with the LUCA. People argue based on these kind of things and say there must be a LUCA, and you know very well the arguments with LUCA being there or not being there, right? There might have been many LUCAs, and a library of them. And it's the same thing with this kind of RNA. There might have been a library out of which RNA emerged. And to look for an RNA-only synthesis defeats that purpose, of making the argument stronger for an RNA world. Because people like, you correctly pointed out here to his question of, what about Matt Pauner's and John's synthesis? And you pointed out very clearly the deficiencies in there, saying this has to do with this and this and this and this. And so that essentially, again, undermines the RNA world if there is no synthesis of an RNA molecule.
Bueno, entonces vamos a iniciar la tercera plática con el profesor Michael Yarus. Él es profesor emérito de la Universidad de Colorado, es biólogo de formación, recibió su doctorado en el Instituto de Tecnología de California, el Caltech, en 1965, donde ha colaborado. Participó en muchos trabajos científicos, su lista de publicaciones es muy, muy larga, todas ellas de gran impacto, y fundamentalmente ha trabajado en las reacciones fundamentales y el papel especial del tRNA y el mRNA en la selección de estructuras particulares del RNA con actividad catalítica o enlazante y en relación a estas actividades con el origen del código genético. También es autor, aparte de los artículos, de un libro, Life from the RNA World, The Ancestor Within. Él es también un entusiasta, divulgador de la ciencia, en la cual plasma sus artículos científicos, pero a un nivel de que todo el mundo pueda entender. Entonces, le damos la bienvenida para oír su plática de esta noche.
Thank you. So I would like to step outside the speech I was going to make to briefly address Ram's talk. So he ended up as a semi-hemi-demi non-believer in the RNA world. Very definitely. I mean, no doubt about it. He's got to it. And I appear in this program anyway as a believer. But what I want to point out to you before I start with my this is an electrifying experience. Before I start the remarks I had planned, what I want to point out to you is that we are actually doing parallel experiments from exactly the same motives. And I can answer some of his questions. And so this apparent antagonism would make a terrific discussion because in the end, we will end up in the same position. So if you would like to direct your questions to that, that would be great. If you would rather take a nap, that would be great too. So my remark about taking a nap is germane because you can actually hear more of me on YouTube. And I'm not really recommending that, But this is an entirely independent lecture on the same topic. And when you put the two together, you will see why I'm working as I'm going to tell you I'm working tonight. And you'll have a bigger picture if you can tolerate 2 hours of my lectures. Anyway, so that's on YouTube. It is independent. You don't need to understand either of the lectures. That one or what I'll give tonight to understand them as independent presentations. Okay. So what I'm actually going to talk about is I'm going to have the effrontery to talk about the origin of life, and I'm going to tell you that I know how it happened. Well, I'm actually not that bold, but I'm going to show you that it's physically, chemically not absurd. In fact, there is a completely plausible way to do it based only on things that we already know work in Eppendorf's in the laboratory. Okay, so the route to telling you that, the route to making that clear to you, I hope, involves first, I got to tell you how I make a model for which I can do calculations. So I'm going to do some calculations, but based on biochemistry that we have measured in the lab. And then I'm going to tell you the calculations are kinetic, but they lead to a molecular picture that I'll call MCU for the origin. And and then you can take this picture and you can simplify it further to an amazing single idea, which to my knowledge nobody has ever said before in biology, which is that templating and catalysis are actually the same idea. And that explains how you could get templating out of the mush that Ram was talking about. Okay, so we need a calculable model. Let me expand the idea of a calculable model first. I'm going to take my gene product to be a 5' 5' dinucleotide like coenzymes. 50% of proteins today, enzymatic proteins, still use RNA to do their chemistry in whole or in part. So coenzymes are an ancient form of catalytic RNA, and that's going to be my gene product. I'm going to show you that we have discovered a new kind of RNA synthesis that is mind-bendingly simpler than what people are trying to do when they try and think. So that's what Ram was addressing, and I agree with him. If you try and address the origin of life by saying, well, the origin of life is trying to think of a way of coming up with ribozymes, doomed. You're never going to be able to do that, as he was in fact implying. You're not going to be able to do it because a ribozyme is a horribly complicated thing. You need to go for something simpler. So I'm going to show you a kind of RNA synthesis that you haven't probably seen before that allows you to make something simpler. And then I'm going to show you that there's a general thing you can say about the way selection alters chemical pathways before you have genetics. And that's very important. If it first doesn't seem very important, and maybe there are things in your life that are much more important than this, even after you understand it, but it is important that you need some way to evolve before you have genetics, because otherwise you could never evolve a genetic system. Genetic systems have to come from the process of evolution, and you can't use genetics to evolve them the way Darwin did. So I'm going to solve that problem with this part of the speech. Okay. Gene product. It's a 5' 5' RNA. It can be written in standard nucleotide notation. It can be written NPPM. It's just like, I think maybe I say that. Yeah, here. It's just like NAD, which you're very familiar with. NAD furthermore, will do chemistry quite independent of a protein. It's composed of an A, like many cofactors, many coenzymes, and then a chemically active residue, which in this case is nicotinamide. So that's going to be my gene product. And the neat thing about it is, the neat thing about thinking of that as a gene product is that this actually has a closed form. It's not the form that you find on many proteins, but this form can be seen, a scene in which you go back through the 5' backbone. Here's one ribose, here's another ribose, here's the A, here's the nicotinamide. That looks like a dinucleotide. It looks like it could base pair. And that is not a hallucination. That does base pair. There is a reaction, very easily identifiable if you go looking for it, in which you put in a 3', 5' normal RNA, you supply activated nucleotides and normal nucleotides, and by base pairing, this thing encodes something that looks like a coenzyme. Okay, so we call this cross-templating, just to have a name for it, a name for something where a 3', 5' backbone, this backbone, encodes, literally encodes, by base pairing of the Watson-Crick kind, a 5', 5' backbone. That's the sense in which you're crossing. So this cross-templating, we have shown, works for a lot of different nucleotides. Well, it works for both AU type and GC type base pairs, for example. And that's because, even though you've never heard of it, it's actually easy. So, and the sense in which it's easy is in this slide. So this is 4 U's. Well, don't know why it did that. This is 4, yes I do. This is 4 U's, and here are 2 free AMPs paired. And the coordinates that I started with in this little model are precisely and exactly those of RNA, normal RNA helices, in which the backbone is the same on both sides. And the observation, the thing I want you to see is that this phosphate oxygen can attack that phosphate without doing anything, essentially. With a little rotation here, you can make a 5' bond from normal helical coordinates with very small motions. And that's why cross-templating is so easy, I think. So here's a summary of the reactions. Now, there's a normal kind of chemical reaction in which the 2 nucleotides, 2 5' phosphates stack, and that can, that can give the NPPM product. But there's also a templated product where very simple in the sense that it's homopolymeric, pyrimidine polymer will encode the same thing as long as these things pair the same way. And so here are the references for the biochemistry. But the thing I want you to remember that we're going to keep coming back to later is that this arrow is red. It is red. My God, I had never expected it to be red. That just means that this reaction is faster. Now, this is a second-order reaction, and this is a third-order reaction. So it's a little, it's complicated to talk about in English, probably also complicated in Spanish, to talk about the relative rates, because they don't have the same units. But in the test tube, if you just add dilute reactants, this reaction will be the faster one. And that's what's going to make all the difference for us in the end. Okay, so this is a question, I'm now going to answer Ram's question, and you may hate the answer, he can tell me in the question period if he does. So we need something that looks like a primordial situation. So in my case, I need something that's explicit enough to make calculations. So I use what I call a sporadically fed pool that receives dilute, undependable amounts of nucleotides at undependable times. And so that's my chaotic system that's supposed to represent the primordial Earth. Okay, so sporadically fed pools. And here we even selected, I mean, the similarity between us runs down to the art we selected for our slides, if you want to ask about that in the discussion period. So here's Michelangelo's famous painting, and I'm using it to illustrate the fact that if you put a chemical system, a purely chemical system, under selection, you can, even though it can't remember what you've done because it doesn't have genetics, you can radically change the chemistry. You can make chemical systems evolve. Now, I'm not sure evolve is the word I want to use because this is not Darwinian in that there's no way to encode what you've done, but still they change under selection. And if you want to read about it, the reference is there. And my screen is going away, but only partially, luckily. Okay, so this thing the change in chemical reactions that are prominent under selection that occurs without genetics, we think it ought to have a name because it's the key to getting a genetic system out of a non-genetic background. So we call it chance utility. And there's a paper about it. Okay, so now, so this is largely theoretical so far and modeling. But I want you to appreciate that there are actually experiments behind this. So I want to talk about the data for a while. So here's a thin layer chromatography, a PEI thin layer chromatography, in which we cook up the nucleotides I've been talking about, and we make some 5' stuff. If you cook for 24 hours, you get an appreciable amount of it. There's an appreciable yield. This is not a very slow reaction. And so there's the APPA product, and here's you can see the hot P32 5' AMP that's in there. But I want to just point out that there are some things you don't see. This system has amazing resolution in that you can see the 2', 5' linked PAPA here. You can see the 3', 5' link, the normal RNA linkage there. And if you cook up for a long time, you see just a trace of the 2', 5' coming up. It's easier to make. But the thing that's really easy to make, and that's surprised me anyway for an unguided reaction, is that you really make the coenzyme-like thing in an extreme majority. Okay, so using those measurements, you can do kinetics, and now I'm going to use those kinetics to prove that if you had sporadically fed pools of cross-templating RNAs, and you had selection for the gene product. The gene product did, like NAD, did some redox chemistry that you could select, so you could select pools that did that better, then that selection would beget inheritance of an encoded chemical transformation. You would switch from being a chemical system to being a genetic system. And that's the burden of my talk. So that's the thing to try and understand now. Okay, so we calculate some pool data. Here's a pool. Here's a sporadically fed pool. The things to notice are, the things that are going to be important are, here through, you can think of, if you want to, you can think of this as days, and here's the molar concentrations of all the reactants. These little spikes are random 1.2 millimolar spikes, so we're in the millimolar range. To some people that's dilute, to others not. You can take your choice. Here are the little spikes of the one unstable reactant. The unstable reactant, of course, is the activated nucleotide, and it comes in and then decays rapidly. Stable things actually accumulate. When you've had enough time for these random spikes to total up to 5 spikes, you've got 5 times as much of a 5' AMP than you put in as your quantum of supply. Okay, so eventually you start getting the NPPM coenzyme-like product, and the thing to notice particularly is here that the temp over chem, the templated product, the templated route to the templated route, which is temp, divided by the chemical route to this, the temp just increases continuously with time. Templating is the favored route. Without selection, it becomes dominant because it's the faster reaction. And that's the thing we're going to come back to. Okay, so you can do all kinds of calculations. You can watch, for example, the distribution. So here's the amount of NPPM, and here's the fraction of total counts, and you can watch as you go from a time of 50 to a time of 100 to a time of 150. You can watch that distribution spread out. More relevant to us is you can do selection. So here's strong selection, the way we do it, just to illustrate that the probability of being selected is going up as the concentration of your product goes up. That's the idea. This is how we're embodying the idea that the product is favored. Make sense? The product enables you to be selected. The product is favorable. And these dots are just individual simulations to indicate, to remind you that a probability of being selected doesn't get you selected unless the probability is 1. There actually is a distribution at every point along the selection curve. And so you can do selection. And you can start out with those distributions I showed you. And when you do mild selection, the mean of the distribution shifts. And if you do strong selection, it shifts more, and the individual points shift. This end goes down, the curve crosses over, and things with more product appear more prevalently. They form a greater fraction of the total output of the pool. Okay, so you may be saying to yourself, why is he saying this? And I just want to show you that everything's working. This is a rather complicated computational task. I just want to show you that everything's working as you might expect it to. Now here's the payoff. Here's the money shot. Here's the templating over the chemical route. Here's the change in that when you undergo selection after the pool is a certain age. Here the pool is young, here the pool is old. Here the change in templating is small. Here the change in templating is large. The point is that in a pool that's dying at a constant probability and that's subjected to strong selection, there is blazing selection for templating. In fact, it's a more complicated calculation which would put you to sleep, but I'll talk about it if you want me to. In fact, this is so dramatic that you can start out with a pool that doesn't template, that as a large minority is making the chemically synthesized NPPM, and in one step of selection, you can jump to majority templating. You can do really dramatic selection with a pool that's young. Pools that are young change their ways, as you can see. This tells you they change their ways more easily than pools that are old. Okay. So these are kinetic results. They are based on rate constants that are measured in my lab. But, you know, these are the things I'm showing you are done by solving the system of differential equations for those reactions. And so they're chemical results. And the bang, for me anyway, the bang really comes when I think of those not as kinetics, but as what molecules are doing. So I want to make that switch now and in 2 stages to make it followable, because this is really the point. So if you ask yourself, what is necessary and sufficient for one of these pooled systems to suddenly become a genetic system, there are, I claim, at a maximum level, there are three things that I call molecularity, catalysis, and utility, and that's why this is the MCU pathway. So you've got a bicycle that's got 3 cranks and moves only when the 3 things are present. Okay, so we're talking about chemical synthesis and templated synthesis, and the molecularity point is really simple, but in the end, utterly decisive. And the really simple thing is that when you have a template, you've got a reaction in which 3 molecules join to make the reaction center. Here, you only need 2 molecules to join to make the reaction center. And that makes all the difference. Another thing, the C factor, is that this chemical route, so chemical route on the left, templated route on the right, this templated route is faster, as we've been saying all along, because the template, as I showed you with that molecular model, the template can bring the reactants together in a conformation in which there's only a short step to the transition state, so that the product is produced faster. So that's why this arrow, there's another red arrow, this is why this arrow is red. In fact, this is the same scheme as I showed you before when I talked about that. So that's the catalysis step. And there are steps beyond that, which we've called here chance utility. There are steps beyond that that have to do with the accumulation of reactants in pools. Pools actually strongly stimulate these intrinsic qualities of the reaction. So the reaction's arrow gets even redder and even bigger. And so the net result of these 3 things is that if you have a product that does something good for a pool and you select it, the pool itself will switch from the chemical pathway to the templated pathway. Okay, so this is all, this is all explicit stuff in the computer. So it's really amusing to explore this world. And it has this world of the pool, the periodic pool, and it has some amazing properties which you wouldn't have expected. For example, I'm now going to show you why the pool evolves faster if you die of some source, some death. You die some death that has a low constant probability, like you're hit by a bolide coming from space, and that annihilates you and your product and your descendants. That's something that has a constant random hazard. If the sun comes up every day and evaporates you, that's a periodic hazard that occurs, it's a 24-hour cycle. And those things are evolutionarily fantastically different. So here's one way to see it. So here are 1,000 pools that perish from the periodic sunrise kind of hazard, and they're allowed to run for 50 lifetimes, or 50 days if you like. And then what I've plotted here is, I've plotted the amount of dimer output, NPPM output, versus the templated route over the chemical route. So this dotted line is actually the least squares line through these 1,000 points. And you can see that rather strongly, this kind of pool perishing periodically, as it increases its output, it's getting it more and more from templating. Okay, makes sense in terms of what I've told you so far, I hope. But here, if you're perishing from something like a random hazard, something like an impact, here is 1,000 pools again, and the dotted line is the same in the two graphs. And what you see is that if you perish from a constant hazard, there's this huge limb of high output pools, which also are templating like crazed weasels. So that's why perishing from this kind of constant random difficulty, whatever it is, leads to fast evolution, because you have these high levels of product, and if you choose those high levels of product, you will choose pools that get high levels of templating, so you'll switch to templating even faster than I'm telling you. Okay. So just reviewing some properties of this system. It's quite simple. Only the small homopolymer and the nucleotides are required. Only 2 complementary nucleotides are required. This obviously works with U, A, and A-like thing that's reactive, so you don't need G and C. You can do this with 2 complementary nucleotides. I've said, though I haven't shown you, that millimolar ribonucleotides are enough, given the stuff we've measured in the laboratory about how well these go. It's actually stimulated by chaotic conditions that you might have on warm rocks on an early Earth. That's because the chaotic conditions add reactants in every possible order. And that means that you always find the optimal order, because always some pools will have that order. So it's stimulated by, evolution is stimulated by these chaotic conditions. It requires only short times, and that relates to the thing I mentioned about having conditions where you can do 1 cycle of selection. That, at these rates, only requires a few days. So you could have an Earth that does not have genetics, and then whammo, in 1 week, you have a genetic system functioning in a pool, which is faster than people usually talk about. But when you do the actual calculations, that's what comes out. So optimal pools can be selected for function, and if you do that and just leave them alone, they accumulate large quantities. So you can have, from MCU, you can have the ideal evolutionary precursor in which a useful function has been selected, and furthermore, the entity that makes the useful material can accumulate large quantities. Okay, next step and last step in the talk. We can actually simplify, again, the MCU pathway to one thing, which I called at the beginning of the talk, I called template catalyst duality. I think that's what I called it. And that's what this diagram is supposed to illustrate. So here's a template. It's got a couple of residues that have secondary bonds to a couple of things that we will think of either as pairing nucleotides or as substrates. Well, what is supposed to be clear from this, I don't know whether it is or not, what is supposed to be clear from this is that a template is an entropic catalyst. They are the same thing. The template, like indeed the ribosome, does its catalysis by bringing together two things that are reactive, so they react faster. And we can describe this big blue thing as a template, or we can describe it as a catalyst, and we can think of these as nucleotide pairs, or we can think of them as substrates of this thing as a catalyst. And we're actually performing the same chemistry, but we call it by two different names, and usually these are not discussed together. Anyway, the template in the MCU path is bringing together two nucleotides and catalyzing their reaction. And so this is a yet simpler way of thinking of what's going on. Okay. So I have told you a somewhat laborious story about how you model pools, primordial pools, but you shouldn't hold that against what I've said, because if you think of this as a template speeding the reaction of two things that it brings together, that is absolutely general. That is physically and chemically plausible without any of the many, truly the admittedly many mathematical assumptions I have had to make to bring forth these results, okay? So the idea that a template speeds a reaction is plausible in a sense. If I hadn't done the calculations, I would probably give a seminar in which I would start with that concept. But you wouldn't believe it. So instead, I've gone through this long calculation to show you that it actually works. You can make everything unstable, you can have variable supplies of substrates, and it continues to work. It continues to yield the switch to templating. So without any particular mathematical or chemical assumptions, it's quite reasonable that when you increase some useful product, you will be selected. So it doesn't require the mathematics, is the point. So selection of sporadic pool chemistries can easily create a primordial gene expression system. It's how I suggest maybe we could make the step using known RNA reactions to the idea and the function, most of the functions of genes. And if there are any questions about that, I'd be happy to try and answer them.
Perdón, algunas preguntas. Muchas gracias por nuestro ponente. ¿Preguntas?
Okay, then I have the feeling, thank you very much for your nice talk. I have the feeling that you're saying that the natural selection was before the other sorts of processes in the Darwinian or Darwinist evolution. Then I have been working the last years with people from chemistry and biologists as well, but I have the experience that a chemical reaction is not yielding like an infinite sort of molecules or other structures. Mm-hmm. In my experience, I would like to say that it's somehow related with the fact that we have just one structure for genetical or very complex machinery for this evolutionary complex structure that, or machines that we are. Mm-hmm. But how can you match the fact that chemical reaction is not producing an infinite collection of molecules and just, let's say, a set of them, mm-hmm. and the fact that we have this Darwinist evolution too. Yeah. And another question, is your model with differential equations somehow related with the Leslie matrix? Because anyhow it...
Related to the...
Leslie matrix? Remember in ecology, the matrix of Patrick Leslie?
I don't know any ecology, so I can't answer that question.
Okay.
So you point to a very important quality of the model, which I agree is true. That is, I assume nucleotides are available, right? And they must be partially activated. And the reason I assume that is, for example, the work at the MRC, which shows that there are plausible ways to synthesize them. It is, I think, a mistake to look at something you don't know how to do and say that it's impossible, because you know, your coat is always in the last place you look, right? That's automatic, because then you find it and you stop looking. And so we haven't looked, I think, in the right place for a lot of these reactions. But there are good models for phosphorylation, for the synthesis of nucleotides, from plausible, at least, plausible prebiotic small molecules. And so I say, well, we've got this earth full of chemists who are trying to make nucleotides. What, in fact, could they do if they succeed? And my point, look, if they can give me nucleotides, in a few days, I can have a genetic system up and running. So you're right to point to my assumption, but that's just part of my shtick. I won't feel bad about it.
Can I do another question? ¿Puedo hacer otra pregunta?
Go ahead.
Nosotros, sí. ¿Otra pregunta?
Oh, okay, well.
¿Otra pregunta o no? Si no, pues...
Hi, Mike. Really good talk. I have just a question, just so I can try to understand what you mean. Let's just say I had some random sequences of oligos, RNAs. Some of them are, let's say, self-complementary, just by random chance. And I let this sit on my bench for a while. After a while, only those are left because they degrade more slowly, correct? This is something we know. Would you call that a genetic system?
No.
Well, why is that not a genetic system?
Your system is so humble. I mean, my system encodes a chemical reaction, so that's why I'm more enthusiastic about it.
Yeah, but the chemical reaction is degradation, right? Degradation of assembled systems is slower, so how is this not a genetic system?
It is one of the reasons I like my system the most, that all those, I didn't emphasize it, but all those components are decaying at literature rates. And so all those conclusions are independent of the stabilities, because the stabilities are there in the differential equations. But I think that selective degradation is really very interesting. I mean, you may remember there are classical experiments that show that when you have a 2', 5' linkage in the middle of an otherwise regular nucleic acid, it has very much increased instability. So decay is important, but I agree. And I've said everything I can think of to say about your question.
¿Otra pregunta? Bueno, si no, regresamos.
Gracias.
Hello.
It's the chemical selection, a sort of natural selection?
Say it again.
It's the chemical selection, a sort of natural selection? How can you do the match or link between the chemical selection and the natural selection?
Are you asking me how I envision selection in a pool?
Or the natural selection at the molecule scale? Is that a sort of chemical selection?
Well, I mean, this is not natural selection because there's no genetics until you've selected it. So that's the point I wanted to make strongly in the main lecture, that this is a way of stepping from a pure chemical system to one that encodes the chemical reaction that we want. So my system is just beginning to be capable. Oh, there's something else I want to say too. My system is just beginning to be capable of natural selection in the usual Darwinian sense at the end of my experiments. I mean, I watch it sail off the right side of the graph, and I wish it well, but I don't follow it into that era. But I want to say something in the spirit of your first comment before. So you should recognize why a lot of people are going to hate this when it's published. I have eliminated replication. There is no replication in my system. My point, and that is extremely heterodox. That may go too far even for RAM, I don't know. That my point is that the clay catalyzed reaction for making these simple polymers is so good that as soon as you had, I mean, clay is the world's most frequent mineral, so as soon as you had activated nucleotides, in the same sense that I'm saying, as soon as you have nucleotides, you have genetics, as soon as you have activated nucleotides, you would have these linear polymers. And so I'm saying you can simplify the way you think about the origin of biology if you're willing to define biology as the origin of an inherited chemical reaction. You can simplify by just setting replication aside. People may laugh at you in the streets and point, but it's logically a perfectly reasonable thing to do in order to get a system that will actually work, as mine does, in a completely chaotic environment. And sorry, that's not your point, but you reminded me of it.
Thank you, thank you.
¿Otra pregunta? Bueno, si no, le damos las gracias al profesor Yari. Quiero agradecer a los tres ponentes por sus excelentes conferencias que nos han animado mucho a seguir todo el resto de la semana. También quiero agradecer a los organizadores de este evento y en particular al Colegio Nacional por ser posible que eventos como este tengan lugar y que fundamentalmente atraigan a muchos jóvenes, que esa es la idea principal. La juventud es el alma de nuestro pueblo y en ellos vamos a depositar la ciencia del futuro. Muchísimas gracias a todos por su asistencia y un aplauso muy fuerte nuevamente a los ponentes de esta tarde. Mañana tenemos también un programa bastante interesante. Van a ser 3 conferencias también. La moderadora es la doctora Sandra Ramírez. Va a estar presente el doctor Müller, la doctora Murel. Son 2 nada más, perdón. Y el jueves también, o sea, el resto de la semana vamos a estar muy ocupados tomando mucho del mundo del RNA, que ya no es el patito feo, sino el cisne muy precioso, como estamos viendo que se está transformando. Pues a todos muchísimas gracias, los esperamos mañana a las 6 de la tarde. No sé si Antonio quiera decir alguna otra cosa. Bueno, pues muchísimas gracias a todos.